Looks good. Okay, so good morning, everyone. Welcome to the third and final day of the Florida Macroalgae Workshop. My name is Darcy Young. I'm the Director of Planning and Communications with the Sarasota Bay Estuary Program. We're looking forward to debriefing this morning after our Wednesday geographic area breakout sessions. And then we'll hear from three speakers about macroalgae management case studies. And then we'll wrap up with the facilitated workshop synthesis activity that I'm really looking forward to. So to get the morning started, I'll hand it back over to our workshop facilitator, Caroline Donovan.
Great. We always start the day with a little bit of housekeeping here. So on the screen, you should be able to see the menti.com website and code to use. So hopefully people are on the Menti slide as well on a different browser on your phone. If you have any Zoom or Mentimeter technical questions or problems, Christine Quigley will be monitoring the chat in Zoom. And if you have any questions, you can chat her privately or chat everyone and she'll respond. If you'd like, you can change your name to just let us know more about yourself. You can put your name, your affiliation, and maybe what estuary you're most interested in. And just as a quick reminder, the workshop is being recorded. So we have a nice warm-up Menti question for everybody. And I'll give you guys a little bit to chance to respond. Okay, people are still answering, but I think it's no surprise. Most people, turtle grass is winning by a mile, and coming in second is halafala, which is just a fun name, and beautiful seagrass. And rupia is last. Oh, sad. But I'll let everybody keep voting on that. We're still getting some answers in, so feel free to keep putting your answers in. Of course, I'm sure there's lots of people that can't pick just one, and it's always great to see a mixed bed as well. All right, so that was just a fun way to get to know how we're going to use Menti. We're using Menti for quite a bit of today's workshop. So the agenda. We are doing the welcome right now. We're going to, yesterday we did or Wednesday, the second day, we did breakout sessions by estuary. And so today we're going to have a report out, a more in-depth report out from each of the facilitators and scribes that were leading the breakouts. And we're going to be sharing screens and using Menti for that as well. We then have invited speaker presentations as well as a panel discussion. We will take a break. And then we're going to do a workshop synthesis and storyboarding activity. So I'm really excited about this. We've done, 3 full days of workshopping, and we want to really think about how we can put that all together and report that out in a nice workshop report after this week. So I'm going to go to the next slide. I believe I'm going to stop sharing, and then Darcy is going to present, or Jay is, I believe. Is Jay there? Yep.
I think we were going to kick it off with Gary and Chris at Tampa Bay if they were ready to go first. What do you think, guys?
Yeah, we're ready.
I'll swap out this Mentimeter. Sorry about that, guys.
That's okay.
So we are going to go ahead and brief out kind of the discussion that we had. I'm going to show our Mentimeter results from yesterday. So hopefully you guys can see it. We had a series of questions that we asked. We had, I don't remember, maybe 16 or 17 people in our group. So we had a relatively small group, but we had a really wide breadth and depth of folks that were on the breakout group. So it was actually a really, really interesting discussion. I think the worst part was just that we ran out of time. So, but we did cover most of the questions, and some of them kind of bled over to one another. So I'm just going to go through them one by one. And Gary was my scribe. So Gary Rowlerson, if you have anything to add before we go on to the next question, by all means, please, speak up. That's fine. The first question that we asked the group was the one word that describes macroalgae in Tampa Bay. And I think really the overlying theme in all of our discussion the other day was that we have a lot of questions, which is really awesome that we kind of landed on that first, the idea that macroalgae in Tampa Bay are relatively understudied, because that's one of the overarching themes of this workshop, was to identify those knowledge gaps and those data gaps, and then come up with a plan moving forward. So understudied was definitely the bigger word that came up when we asked that question. But you could see kind of all of these are dancing around the same idea. I did like the one that said annoying. I don't remember who wrote that, but that was a really good one too. But that sort of brought us to the next question, which is, what are some of the knowledge gaps in Tampa Bay, if any, that we've noted from the workshop? And so we had a lot of really good responses for that as well. One of the things that we sort of discussed as a group was the idea of what sort of habitat implication does macroalgae. Specifically, we were talking about attached algae, so Caulerpa proliferans, we've talked a lot about over the last few days. And so some of the questions that came up in terms of knowledge gaps was, well, what is the ecological value of Caulerpa proliferans, especially relative to seagrass meadows? Also, some of the temporal and seasonal variability in the grass, when is it most abundant? We saw some really good talks about that. And fortunately, Roger Johansson was in our group. So we were able to kind of pick his brain a little bit more on some of the stuff he presented on, I think it was Monday. And we also talked a little bit about a workshop that was done back, I believe it was in 2002, and Roger was actually part of that workshop, and it was done by, I think, the Maryland Department or Division of Natural Resources. And that was the topic of that workshop. It was mainly focused on I believe the outer banks area of Delmarva, but it was really good to kind of tie in some of the work that had been done in the past to help guide what we need to do in the future for Tampa Bay. Gary, did you have anything in your notes to add to this slide?
You're covering a lot of the good stuff, Chris. Thank you. The only thing I'll add is that we also had some discussion about what are the wildlife implications as we see these transitions between macroalgae and seagrasses. Are there differential responses for fish or manatees or primary, secondary consumers? What are the implications for the different wildlife that are in these systems?
Thanks, Gary. Yeah, we did also touch very briefly on the role that sediment might play, not so much on driving out macroalgae, but more the interplay between seagrass, attached algae, and how that sediment variability may play a role. So a lot of good stuff that we talked about there. When we went on to the next question, which is any ongoing research, there was some pauses, and we sort of hit on some of the highlights. But as you can see, we didn't really, there aren't all that many that are specific to filling the knowledge gap of macroalgae ecology in Tampa Bay. And some of the ones that were brought up were ones that I think are consistent really across the West Coast. So the Swiftmud seagrass mapping, there are seagrass transects and fixation monitoring programs that are done throughout, and then fisheries independent monitoring that FWRI does as well. Gary, did you have anything on this one to add?
Thinking about the transect work that we've been, basically Roger started in the late 90s. well, one of the things we recognize is that there is some algae information in that database, but it's kind of sporadic, and so we'll be putting some ideas down over the next several months before we do the next round of training to see if we can improve the reporting on that for that set of work.
Yeah, and even though, you know, we had relatively few answers to this question, as a whole, the Tampa Bay estuary is, as I would say, very well monitored, and there's a lot of folks that are keeping the pulse of the estuary. And so I think, as Gary said, we'll talk about in the coming months how we might want to fine tune some of these monitoring efforts in order to get at some of these specific knowledge gaps that we talked about. This one kind of bled into the previous one, so we didn't really have a whole lot of time to get to that one, because we talked a lot about monitoring in the previous question. But we did talk about this one quite a bit, which was the idea of what sort of information is out there relative to the sequence of events or that successional pattern that Nate and I talked a little bit about the other day, looking at imagery and seeing how things kind of move from one ecosystem to another or one habitat type to another. So I thought we had some pretty good responses here. Anything else, Gary, to add to this one?
Well, just to reiterate, thinking about the imagery work that y'all have done for the last 3 decades now, being able to dig into that a little bit further, I think will help us tremendously along these lines.
Yeah, and I think specific to Tampa Bay, that middle top box was really a great question, which for those of you who may not be familiar with Tampa Bay, in the old Tampa Bay segment, which is up in the northern western part of the Tampa Bay estuary, we've been seeing persistent phytoplankton algae blooms in that system, and there's been a lot of discussion really led by the estuary program, but also just among the various research groups is what might be behind that. And so that was a really good question that somebody had put on there, that, you know, specifically what's the role or what's the dynamic between, you know, phytoplankton blooms, persistent blooms, and this transition, this apparent transition from seagrass to macroalgae. So it really quite a bit of good discussion there. And unfortunately, that's kind of where we ended. We got kicked out. So we didn't really have time to talk about the historical perspective. We did throughout our discussion, but a lot of folks that were in our group, some of them had been working in that system for decades. And so it was really nice to hear some of that historical perspective on what macroalgae blooms looked like in Tampa Bay back in the 1970s and 80s. That was sort of the low point in terms of the health of the estuary. And, you know, we had folks like Roger that was in our group that really could give us a good snapshot on what the system looked like, and actually collected the data like Gary Rollerson had indicated. You know, the Transect data really started with Roger and Walt Avery at the city of Tampa back in the 70s. So that's a really long-term data set. So we do have a lot of really good historical perspective. Unfortunately, we ran out of time, so we didn't really get a chance to go in too deeply with that. But I would say, and Gary, I'll hand it over to you for any last words before we wrap up our group. But I would say by far the overarching knowledge gap that was identified early on in our discussion was the idea that macroalgae is largely understudied in the context of these estuaries and Tampa Bay specifically.
I'll just agree with everything that Chris has said, and thank him for wrapping it up. I guess the only thing I will add is that over the next several months, the estuary program will be digging back into our research plan for the next several years as part of our 5-year update to the CCMPs. And so it was the last one was done it came out in 2017. Time is flying, but we'll be working with the TAC over the next several meetings to update our research plan, and I'm sure this will be a considerable component of that. Thank you.
All right, that's it from us.
Great, thank you guys. So Darcy, you should be able to share your screen if
Yep.
Ready? And I guess I'm going to challenge you to keep it to about 4 to 5 minutes.
I think we can do that. What do you think, Jay?
Yes, we can.
Okay. All right. So I'm sharing our word cloud there, and you can go ahead and kick it off.
All right. Well, thanks. I want to thank Darcy also for, on top of all of her duties, coordinating this conference to also be a scribe for us in our group. And Darcy, do you recall? I don't have the number. How many participants we had in our group?
We had 26.
26. That's what I thought. And I'm sure you'll see probably a bit of similarity between our discussions and those of Tampa Bay. A lot of the issues and knowledge gaps are pretty common to both these estuaries and perhaps some of the others, but there are some unique things that we can highlight from Sarasota Bay. And interestingly, our group noticed two things. That most importantly is that we're noticing that algal biomass, the drift algae, is increasing in our bay, and even some of the cyanobacteria as well. We're noticing increases in the abundance of it, and that it's a problem. And we ought to be doing something about it. So we had a lot of invested and interested and concerned people in our group. So not surprising, and that's good. So in terms of knowledge gaps, we had a lot of responses, kind of looked at them and broke them down into some major categories. And one that came to light was the fact that what are the impacts of nutrients? We are really laser focused on identifying our sources of nutrients, how they are attenuated or effectively available in the estuaries, what sources are more important than others. So there was a lot of focus on not just the sources of the nutrients, particularly nitrogen, but what type of responses are we seeing in our bays. We also recognize that if we ever do some sampling, and there is some sampling and we intend to do some more, that we ought to be looking at seasonal considerations as well. A lot of people say, and it's limited by our efforts collectively, that a one-time snapshot a year, while it's good information, may not be enough to help us understand the ecology of these things. And then also some successional patterns of macroalgae over time. It kind of plays into that. We won't know about the seasonal aspects of it without being able to monitor it in some capacity more than just once a year. Then we had an interesting participation from Joy Jackson from DEP because we were looking at the relationship to the nutrient criteria that were developed for our estuaries, and in light of these new issues that we're seeing with increasing macroalgae, in addition to declining seagrasses and other ecological response mechanisms. So that was really good because we were able to tell her that our program, in fact, is looking at reevaluating our nitrogen criteria to give us a better understanding of being able to detect in our nitrogen standards these issues that heretofore the nitrogen levels have not really been able to coincide with the declines in seagrass and the increase in macroalgae. So that was really encouraging because that's going to be a focus of our estuary program over the near term. And finally, the role of climate change as a driver. Obviously, that's an underlying thing for all of our estuaries as climate change continues to march forward, how it's going to impact what we're seeing today. The next one, Darcy.
Yeah, thanks, Jay. And before we go off the knowledge gap slide here, you'll see on your Mentimeter that we asked you a ranking question for those 5 knowledge gap issues. So anyone on the call, whether or not you were in the Sarasota Bay group, feel free to go on in and rank those knowledge gap issues or prioritize them while we keep doing this report.
Oh, thank you very much. Yes.
Sure. Let's see.
Definitely welcome and encourage you and value your feedback. With respect to monitoring, data, research, we really have just one existing program, and we're very thankful that they've been doing it for such a while, for years now, and it's providing good information, at least for Sarasota County waters, and that is the Sarasota County staff are out there, not just doing water quality data, but they're seagrass monitoring, which includes a good healthy component of macroalgae biomass estimates and coverage is very, very helpful. You may have heard that we are going to be implementing the Eyes on Seagrass program instituted by UF IFAS, and Betty Stogler and her group in Charlotte Harbor. We're going to be bringing that road show to Sarasota Bay this year. So that'll be another program that can help us. Gaps in our data. Again, expanding monitoring seasonally so we can understand the dynamics, the temporal dynamics and the spatial dynamics of what we're seeing a little bit more clearly. I have a note from Darcy, nutrient sources, reclaimed water application. We're looking at understanding how the impacts of those applications as they enter our waterways may be impacting increases or blooms of macroalgae. Did I miss anything, Darcy?
I think that was it, Jay. Thank you. So Caroline, I think you can show our results slide here, see if folks have had a chance to do those prioritizations, and then we can hand it off to CHNEP.
All right, are you guys?
I have one other point in terms of sequence of events that I'll just mention that you know that our group has understood is that, and Dave Demasco has alluded to it with other trends as well, but we see different processes happening in the larger northern Sarasota Bay segment, the open bay segment, if you will, and our smaller embayments to the south, Roberts Bay, Little Sarasota Bay, and Blackburn Bay. So we're seeing different trends in a lot of things, including macroalgae. And also we recognize that there is a real kind of a shift in our systems around 2012 and 2013. So there's something that's been happening in our system around that time period, and we're seeing different responses in our different bays. That's what I want. I wanted to mention that as well.
Great. Well, you can see here the rankings are still going on, and it's kind of neck and neck after the first one. So that's interesting. But people can keep answering, and we'll go on to the next group.
Thank you.
So are Jennifer, or is Jennifer or Nicole going to share?
Yeah, I will share my screen.
Great. And if you want to just let me know when you want me to put your Mentimeter question up.
Okay.
Or unless you're going to do it.
I have everything. I summarized it all on these slides from our previous Mentimeter.
Yeah, and then if you want to do the Mentimeter question
okay.
Carolyn, if you just want to have that go simultaneous, that's fine.
Yeah.
Okay, great, thanks.
All right, great. So yeah, thank you all again. And I'm going to just summarize the Charlotte Harbor area estuaries in the next couple of slides. As Jay mentioned in his, we have a lot of different estuaries with a lot of different hydrological inputs. So we separated out some of the results that we got by those systems. And so here you can see we captured some of the historical context for Donna and Roberts Bay, as well as some of the more recent context for succession, what's been going on there. So you can see in the 80s, it looks like the system was a little bit healthier. And then there is this area obviously receives an increased freshwater source coming through Cowpin Slough, which has changed the hydrology of the area. There's been a lot more filamentous algae in Donna Bay since 2018, and lots of sites with algae, and also with Caulerpa starting to bloom in the nearby Lions Bay, which is just a little bit north of that. You can see on the map there, and Donna and Roberts Bay as well. So moving on to upper and lower Lemon Bays, you can see here we've got historical context at the top and more recent events down here at the bottom, and we appreciate everybody in this breakout group. I think we had almost 40 people in our breakout group providing lots of information. So trying to capture it all here. Basically, there historically it looks like there was increased attached algae and algae blooms over the summer. People said that upper Lemon Bay was a little bit more haliduli dominated. When we're talking about seagrass, lower is a little bit more mixed, closer to the pass. More recently, the red tide hit this area pretty hard. It looks like there might be a large residency time here. And it looks like there was a blue-green algae bloom 2019, a little bit more of those attached algae, green algae in 2019 as well. increased caulerpa in Upper Lemon Bay. There was a decline in halidulysine in Upper Lemon Bay as well. I think increased sedimentation up there also happened after Irma. And drift algae in the water column and increases in macroalgae pretty much all over Upper and Lower Lemon Bays, as well as seagrass losses. Moving on to the tidal Mayak and Peace rivers. Again, there was never any filamentous algae reported in either of these areas from basically when the FIM monitoring began from 1989 through 2011. Then again, around 2012, we started to see an increase in macroalgae in these areas, scallop and seagrass loss in these areas about that same time frame. So more recently, there's been a lot of algae blooms near Hog Island, which you can see here. I don't know if you can see my cursor in the Mayak cutoff area, Alligator Bay up here. Filamentous algae on the Punta Gorda side, which is over here closer to the Peace River side inputs, seagrass losses and boat scarring, green filamentous algae blooms in 2019 and 2020. We're seeing a lot more caulerpa as well in these areas. Charlotte Harbor, main Charlotte Harbor, Gasparilla Sound, Cape Hayes area. Historically, there was always historically a little bit more of that seasonal drift algae, sedimentation in Upper Harbor and Bull and Turtle Bays after Irma. And then again, we've seen a lot more seagrass losses after the red tide event, a lot more caulerpa, a lot more of the green filamentous algae blooms as well. Red drift algae seen in Bokeelia, Cape Hayes, Turtle Bay, and these are the areas that are actually closer, you can see here, to Boca Grande Pass. And again, green filamentous algae in Turtle Bay up here. And higher densities of the drift algae. Even that seasonal drift algae, we're seeing higher densities of it. So Pine Island Sound, Matlacha Pass, San Carlos Bay, and sorry about that, Caloosahatchee got a little cut off in my photo here, but historically, a lot high densities of drift algae in the wintertime again, especially in the Matlacha Pass area. But there's been an increase in attached algae, seagrass species shifts after Irma. I think these areas, it looks like from the comments, these areas are having a hard time recovering from the Irma, followed by the red tide, as followed by this macroalgae. And there's a lot of boat scarring in these areas, really shallow, especially Matlacha if you get out of the channel. More recently, Matlacha Pass has seen an increase in algae in the northern parts of the pass, green filamentous algae, tumbleweed algae, Pine Island Sound, increased drift algae. And you can see Pine Island Sound has actually inputs from 3 different passes here, which is historically a lot of seagrass there too. San Carlos Bay, red macroalgae along the beaches. And again, the Caloosahatchee River, as you may be familiar, has a lot of the cyanobacteria blooms. And finally, Estero Bay, historical context. There's been a lot of disturbance by boats. It's a really shallow area, lots of sediments in the water column. There was a large bloom in the spring, almost yearly, lots of SAV loss in the 80s down here. So seagrass abundance was high in the 2009 to 2012 area, and they've seen a decline in seagrass around that 2012-2013 timeframe as well. Again, seagrass shifts to algae, red algae blooms. So red algae and green filamentous algae both have been seen here, as well as Limbia lights. So that's sort of the historical context of our estuary systems. And on these couple of slides, I'm going to just show what we captured from participants as far as data gaps in monitoring. And I just wanted to give a shout out for the data that we do have when we were capturing that. We've got a lot of great seagrass and algae data because of ongoing programs like our Aquatic Preserves program, because of our Fisheries Independent Monitoring program, and because of that Eyes on Seagrass macroalgae that Betty Stogler and Sea Grant started, we captured that 2018 sort of bloom as well with that citizen science volunteer. Our results sort of reflect what we've been hearing from Chris and Jay as well, is that we need more spatial and temporal information on macroalgae. We'd like to examine it along with the water column and seagrass data as well. And then there were a lot of questions about, aside from that, the relationship of macroalgae and sediments, of climate change perhaps too, also of the inputs, where are the nutrient source inputs? Are we looking at groundwater? There's a few small studies. Do we need to do some broader research with groundwater inputs, hydrological inputs, circulation as well? Those were some great notes that people included. I'm going to switch to the other slide too. So these are just a couple of the screenshots of all the information that we captured. But and again, there was also questions about what the impacts of algae are. Are there macroalgae mitigation strategies? What are the benefits of macroalgae? And again, talks about nutrient inputs, what we can do to reduce the impacts from that. And part of that is the algae speciation, understanding what are the nutrients that are feeding these algae as well. So a lot of great thoughts in all of these areas as well. And we really appreciate everybody's input. And with that, Jennifer, did you have any other notes that you remember from our breakout group?
No, I think you covered it. Thanks.
All right, guys. Sorry for talking fast. Hopefully you covered everything, and appreciate everybody's time.
Thanks, that was great. And a lot of people are responding on the Menti. So go ahead and switch over to your Menti and make sure you get your answers in. And I'm gonna take over that if that's okay, Nicole, just to show people really quickly. And up next is Chuck with the Indian River Lagoon, but here's some of the quick results. So everybody can go on and answer these questions, and we'll be synthesizing all these results afterwards and sending them out. All right, Chuck, are you ready?
I think so. You
Great.
see the slide?
Yes, one word,
Okay.
you see.
So we went through the various questions. These were the top 5 words that showed up in the word cloud. Basically, I think people were mentioning the kinds of things that make us always say that what makes the lagoon special also makes it vulnerable. Knowledge gaps, categorize them kind of 3 main ways. One is sort of how macroalgae see the world, so the audicology of the algae themselves. And then the synecology, how does the world see macroalgae? Where does it fit in? And then people were also concerned about targets for management in the lagoon. So if you're going to manage macroalgae and so forth, what are you aiming for? Ways to fill some of these gaps, the encouragement for keeping this kind of interaction going across the various estuaries, look a little broader afield, raise macroalgae from what typically is bycatch to something you focus on. Add the concept of surveying macroalgae to any kind of assessments that are going to be ongoing, and no surprise, more dollars. In terms of data gaps, similar to what others have said, enhanced spatial and temporal sampling, looking at where nutrients come from, looking at seasonality, but also some more understanding of the species. Species seem to matter. Toxicity is a concern, and how they use nutrients. And then the interactions, again, the interactions with the food web and the other primary producers. In terms of observations that people put either on the Jamboard or in the Menti session, there are changes that seem fairly widespread in the recent past, a variety of different things going on in different parts of the lagoon. And historical perspective, some people saw a signal in terms of increasing stuff. Other people saw some noise because it tends to be highly variable. Almost everyone agreed that we need to have a better handle on what the roles are for macroalgae. Back to you.
Wow, that was oh, my video on. Yeah, that was great. Thank you. I really appreciate that all the report outs are sticking to their time and stuff. So I agree. I am going to move the Mentimeter over to an Indian River Lagoon Menti question while I go through the slides from our breakout. So there were 2 Indian River Lagoon breakouts, and so Chuck just did his, and I'm going to go and share the results of mine if I can figure it out. All right, are you guys seeing the word cloud? Hopefully. All right, so in our breakout, we had about 25 people, and we got started right away with the word cloud. And so I think a lot of what you're going to see is going to come up in the other has already come up in the other report outs. But to describe macroalgae in Indian River Lagoon, the big word was abundant, variable, and challenging. I thought some of the other words were really, really cool, like misunderstood, perhaps, or cryptic. So really, really great responses to the word cloud. We also had a lot of talk about the macroalgae knowledge gaps, and really the data gaps and the knowledge gaps kind of went together in our breakout. And so I think there were some really great responses here, and I want to just kind of summarize or quote someone from the breakout. You know who you are. There's so much we don't know, and there's so much we need to know that fundamental research and monitoring is needed. So here's some more responses about the knowledge gaps. And then, are there any ongoing research and monitoring to fill these knowledge gaps? There was quite a few answers about what is already going on and what's monitoring and coming out with the different surveys and different efforts by different agencies. And so Daniel had mentioned that the Indian River Lagoon projects throughout the monitoring projects throughout, you know, people are starting to coordinate their efforts. And so really the monitoring is happening, but it's more about coordination and working together to fill data gaps. And there was a little bit, we kind of combined these answers, the data and the knowledge gaps. So those kind of all go together. Do you have any information about the sequence of events? So there was some really interesting questions or answers here, but it was a pretty short response. I think maybe a combination of the different Indian River Lagoon breakouts might help kind of answer these questions more about what are the sequence of events that you're seeing, but obviously, you know, excess rain causing nutrient pollution, causing blooms. And sharing 2 sentences about your historical perspective. I think there's some really good stories in here, like changes in species compositions over time and larger, more intense blooms. And I like this. When I began working in the Northern Ireland Lagoon, I witnessed very little caulerpa compared to other macroalgae and seagrass. Now caulerpa is taking over. So I think these are pretty common responses throughout the different estuaries. So I'm just going to leave it there. Do you, Daniel, want to chime in on anything that you heard during our breakout? Oh, and I wanted to also highlight a question from Dennis asking the question, why isn't there more macroalgae on bare spots in the lagoon? We just don't understand their pattern yet, the patterns yet. So that was also interesting perspective of, sure, the seagrasses have gone away, but there's a lot of bare spots, and you would think macroalgae would have come in and taken over, but maybe they're in a transition, who knows? Okay, I'm going to switch back over to the Mentimeter. So there's some questions here. Prioritize the data gap. So what do we want to really focus on? I think there's always going to be data gaps and questions, but what can we focus on? So how the shift from seagrass to macroalgae affects lagoon resources is the top one, and it kind of goes down from there. So thanks for responding to that. And just gonna pause there, stop sharing my screen. That is the end of the report out. And we're going to transition into our invited speakers. So first, we're going to hear a talk from Dr. Jamie Vaudry from the University of Connecticut. She is a scientist looking at macroalgae in Long Island Sound. So Jamie, take it away.
Great, thank you. Let me get my screen sharing going here. Few more seconds.
All right, looks good.
Excellent. So thank you all for inviting me here. As Carolyn said, I'm from the University of Connecticut, and I work primarily in Long Island Sound, in the Long Island Sound study. And as I was prepping for this talk, I really wanted to tell you about some of our success stories in Long Island Sound, but realized that, that I really didn't have time for that. So if you want to ask me later, that would be great. But I'm going to focus today on 3 different techniques that we've used for monitoring macroalgae in Long Island Sound. So before I get started, I just want to mention this has been probably 20 years of work that I'll be breezing through today, and it's been funded by many different sources, and we've had a lot of people participating in this work over the years. So I want to start off by talking about stressors. You know, nutrients are what we identify as the main problem of the macroalgae blooms, but those other stressors include temperature, which I'll touch on momentarily, and then also the presence of invasives. In Long Island Sound, we're finding that our gracilaria species, which are shown here in the upper right, are largely replaced by this invasive species. And we're also finding that some of the ulva have been replaced by invasive species. So we've lost our ulva lactuca, and we're seeing different species of ulva. And this presents a problem or presents an opportunity for the macroalgae, because these invasives are often more vigorous. They're able to do better in cold waters, so they may exist in the winter. They're able to weather the warmer weather during the summer better than our native species. Or they may do better under all sorts of conditions. They're better at taking up nutrients. They're better at dealing with hypoxia. There's a lot of unknown there, but that invasive presence kind of compounds the impacts that we see in these macroalgae blooms that are occurring. So Long Island Sound is, we call it the urban sea, and that is largely due to New York City, which is the bright spot shown in this image from space over on the left. But the corridor from New York City to Boston is one of the most densely populated in our country, and that sets up a situation that is just ripe for macroalgae to bloom in overabundance in Long Island Sound. We see a pollution gradient with higher pollution problems over by New York City and fewer over in the Eastern Sound, where Long Island Sound exchanges and is flushed by the Atlantic Ocean. However, in our embayments, which are shown here as the light blue water bodies, we don't see that same trend. So each embayment reflects its local watershed. That's what these colors are indicating. They're showing us the local watersheds of each of our approximately 100 embayments. And I'll mention right now, people always say, well, why'd you skip embayments on Long Island? In the areas where there are no embayments along Long Island, those are areas that don't have embayments. It's just open coast. So we're going to zoom into Little Narragansett Bay right now, over on the east end, which should be our best water quality. But what we find is a huge bloom of 2 species in the Cladophora family. This is putting down the anchor. You pull it up, and your anchor is just coated with this seaweed. In places, the seaweed is about a foot thick to 3 feet thick. So this is a serious macroalgae bloom. When we were doing some embayment characterization back in the early 2010s, so we were going out to our embayments and seeing what the water quality status was in these systems and how things were doing. And that had not yet been done in a systematic and widespread manner throughout Long Island Sound. As part of that, we found this macroalgae bloom and realized that, that this was far more widespread than we had previously realized. This student here, Amanda Dostie, applied for and got a grant as an undergraduate to look at the coverage and biomass of Cladophora in Little Narragansett Bay. And this really set up how we were going to monitor and evaluate seaweed in these systems. So playing in the background is a video that we get from our monitoring of these systems. So we're looking for percent cover. And what we've done is we have a GoPro affixed to a pool pole, and you bounce along the bottom, taking video of the bottom, and then a person goes through afterwards, extracts still images, and then puts a 100-place grid over it and gets the percent cover in that image. The results are shown in the figure here, and this is a figure from 2014. The red is 100% cover, and the green is about 25% cover. What is happening is Pawcatuck River, and I'm not sure if you can see my cursor, but I'm pointing to the Pawcatuck River over on the right, has 3 wastewater treatment plants on it, and it delivers nitrogen down out of the mouth of the Pawcatuck River. And that water can go out to the Atlantic Ocean, or it can turn the dogleg into Little Narragansett Bay. And we can see the trace of that nutrient impact on these seaweeds and their coverage in this system. In addition to this video survey that we've done, which is, we got, I think, probably 1,500 images in this system to make this plot. It's relatively quick, relatively easy to do in the field, and then requires someone to do some sampling afterwards. We also did grab surveys to get at biomass of these systems, and that's what this plot is showing. So in this plot, the blue areas are patchy, green and yellow are about 1 inch thick of seaweed. The red tones are getting about a foot thick, and at the mouth of the Pawcatuck River, or at the near shore, where it hits the embayment, we had about 3 feet of algae in that system. So to do the biomass surveys, and in this case, I have little black dots which show every place we did a collection. Grabs were great for most of the system, but once you get into seaweed that is more than in a few inches thick, it pops out of the top of the grab. And so in that case, we had to send down snorkelers who took a meter stick and measured the depth of the seaweed, and then collected a known volume to get at the biomass in these systems. So we have a huge biomass in this area. This method of monitoring really gives us a good estimate of the coverage and the biomass. The biomass is far more labor intensive. The other survey maybe took us 2 days. This one, I think we were out there for 5 days total, working on getting biomass between the snorkeling and the grabs. This data is important for us because we're using it to verify models that are now being run for each of the embayments in the Long Island Sound area. That early work, so that was in the early 2010s, 2010 to 2014. Out of that early work, it developed into a desire to monitor our bays and harbors. Coupled with that was a desire to have a report card for Long Island Sound, both the main stem of Long Island Sound and and for each of the bays. And in fact, Carolyn was involved with that. The Long Island Sound study, Mark Tedesco brought in the University of Maryland Center for Environmental Sciences and Integration and Application Network to initiate the report card process for Long Island Sound. As part of that, we realized that our bays were not being monitored in a consistent manner, and that led to the birth of the Unified Water Study. And the Unified Water Study is a supported program overseen by Save the Sound that monitors water quality in all of our bays, or not all of our bays, but about 38 out of our 100 bays throughout the system. We monitor water quality in a manner that is meant to be representative of the system. So groups are out there May through October, every 2 weeks, sampling water quality at stations throughout the embayments. But we wanted to include macrophytes in this assessment. And macrophytes, as you all know, are labor intensive to monitor and hard to get a handle on the variability. So they can be very variable within a system and through time. And so that requires a whole different setup. So we thought, well, what can we do that will tell us something about the status of macroalgae in these systems and be useful for our report card effort that our community scientists can do? Because in this study, these 38 bays and harbors are being monitored by about 22 different community groups. What we came up with was rake tosses. And in addition, we're not just randomly tossing rakes at all of our stations, we're asking people to look for the worst place. So part of setting up a news site is going out and scouting and finding the place where in your embayment that has the most seaweed. If your embayment doesn't have any real seaweed problem, then we'll see that. So you're monitoring 3 stations that have the worst incidence of seaweed. This survey is conducted only in a 3-week period at the end of June through early August, end of July through early August. And we also ask that groups go out 3 times during that 3-week period and visit their stations. So again, we're getting the worst example of seaweed in the system because we're also hitting it at what is traditionally the highest growth period for these seaweeds. So when we, this is leading into the grading system. So these are the 2020 Long Island Sound grades. The main stem of Long Island Sound does not include seaweed sampling. And that's the big green and yellow and red areas in the middle of the sound. And then each of the little circles around the edge of the sound is an embayment or a section of an embayment within that system. What we find is that our embayments, again, do not match. You cannot predict the water quality in embayment based on the water quality in the main stem of Long Island Sound, because these bays are responding to the nutrient loads from their local watersheds, as well as to stressors like restrictions to flow that occur in many of our bays. In general, though, we see that our embayments in the west tend to have lower grades. Our embayments in the east tend to have higher grades. Now I'm going to switch the figure here and we're going to look at just the seaweed grades, because in our embayments, we grade 5 things, 2 different oxygen indicators, chlorophyll, turbidity, and seaweed. So these are just the seaweed grades. And again, we're seeing that you cannot predict seaweed grades based on, the main stem of Long Island Sound. We also find that if you look over at the southeast or the left side of this map, there's a fair bit of green. Down in those areas near New York City and along Long Island, we have a combination of 2 things going on. So along Long Island, those are our deepest embayments. Light is probably not reaching the bottom in those systems, and so macroalgae is not as much of a problem in those deeper embayments. Over by New York City, we do still have some deep embayments. We also have high turbidity and high chlorophyll. So again, that seaweed is going to be light limited as we move over into the Western Sound. So how do we do it? Our approach is that each embayment has at least 3 seaweed stations. Again, they're looking for the worst spot. They're going out and doing these rake tosses on 3 different dates within a 3-week period. So we're kind of capturing a snapshot of what things look at the worst time of the year. We ask our community groups, so this is one of those things where we really needed to think about what can we ask of our groups, what's really important. You know, we could lay out transects and get percent cover in those transects, but that's falsely quantitative in the sense that we really can't ask these groups to do enough surveys or enough rake tosses to be able to say, yes, we have adequately sampled that system. So remember that we're problem tracking here. We then ask our groups to categorize these as none, some, or lots. So they do 6 rake tosses at a station, and it gets characterized into these 3 classes. There's a technical reviewer who then goes through and sees whether they agree with the designation of none, some, or lots, and then also adds 2 categories. So some of the lots get converted to enormous, and some of the some can get converted to minuscule. So there's some rearranging between some and lots, some kind of baseline rearranging that our technical advisor does, but then also the addition of these 2 categories. Then the final step is to look at the species that are there and identify beneficial plants. So those are our seagrasses, Zostera and Rupia, and also our beneficial browns, our rockweeds, Ascophyllum and Fucus. So if beneficial plants are there, that's a plus for the system. It's a boon. It's not a negative impact on the grade. This is just an example of the layout of the data that we get. So we've got 6 rake tosses at a station. They visit that station 3 times, and then we get 3 stations per embayment, and all of that gets pulled together into a score for the system that also includes an error associated with it. And that's important. Again, getting back to that idea that seaweed is variable in space and time. How do we quantify that? How do we capture it? How do we compare it? We've done this survey for 2018 and 2019. We'll continue to compare the intra-annual variability and the inter-site variability, but we really can't do that, and we don't feel like we can do it until we have at least 3 years. 5 years would be better, and really 10 years would be great. But we're going to start looking at some of those intra-annual variability metrics using the 2020 data, and 2021 will be a boon to that. So in general, the comparison for intra-annual variability, you know, it falls along the one-to-one line here. So if you're a high system in 2018, you were probably a high system in 2019. There are some sites, so we have some, you know, that got a B in 2019 and got an F in 2018. So that variability is going to be important to characterize as we move forward. So our third method that I want to touch on is done in the Niantic River. And I noticed in the Mentimeter report outs, there were some suggestions to capitalize on existing efforts. And that's what this survey does. So the Millstone Environmental Lab goes out and they are trawling for larval fish. So they have a particular toe. That toe is always the same length and in the same place. And they go out every, I think, 2 weeks or every month for a long, long time. And in 2011, they started, they said, you know, should we be like gauging our macrophytes? So this is, I'm blanking on the word, but we're capitalizing on efforts that are already occurring. And what they do is they estimate by eye how many 5-gallon buckets they think the seaweed could fill. And from that, we're able to estimate a volume per toe. Interestingly, we had a seagrass decline in this same system in 2012 that was temperature related. And right around that 2012 period, we see the volume of macroalgae really taking off. You know, we see a definite increase over time. Our seagrass is recovering in that system, but unfortunately competing with it is this increase in the amount of macroalgae in the system. So again, an example of an easy way to get someone that's already out there towing to get you some information. So one of the solutions is to obviously reduce nutrient inputs, and that tends to be the focus, as it should, because that is really what is feeding and fueling these blooms. But what we need to consider is also the sources of these nutrients and how we can approach it from multiple angles. So if we can reduce the nutrients coming from our point sources, great. If we can do something to improve septic systems and fertilizer input and stormwater management, again, something we definitely need to do, as that is the source of the problem. But one other thing that we're looking at in Long Island Sound is nutrient recycling or bioextraction. So we're looking at growing aquaculture, growing shellfish and growing kelp and growing gracilaria in an aquaculture setting to look at nutrient removal. But another option is to capitalize on these massive seaweed blooms and use them as a bioextraction process. Those seaweeds could be harvested, they could be composted, turned into fertilizer, and enter, be recycled as fertilizer and enter that food stream. Now, that in theory is great, but in practice, there are some problems with it. First of all, we're pulling seaweed out of often some potentially contaminated areas. It grows near the sediment, which can include heavy metals and things like PCBs and other emerging contaminants. And we don't want that entering our food stream. So there are still questions of, how do we determine that fertilizer created from this composted seaweed is safe to enter the food stream? If it's not safe, how do can we separate it out and put it towards ornamental plants or somehow designate it for other fertilizer recycling uses? The other issue, and I put it over here on the bottom right here, is the need to prevent spreading when harvesting. So as you're harvesting these seaweeds, they can break apart and then go on, and now you have two ulva plants that are going to go and take off someplace. So there is the issue of fragmenting the seaweed and actually encouraging its growth as you're doing the harvesting. But in addition to that, where are you taking it for composting? And confirming that wherever you're doing that composting, you're not going to be spreading those seaweeds to a new system. So those are some of the many layered issues that come when we're thinking about what we can do if we harvest this material from beaches or harvest it from the water. What do we do with it? So, with that, I would say that in conclusion, our systems, we find that the seaweed is widespread. It's in almost all of our shallow areas and very problematic in some of our very shallow embayments. Our deeper embayments tend to be phytoplankton dominated. And really the way that we have confirmed and learned this over just the last 5 years is through support of that community-based monitoring, which has really given us the workforce to go out and get the amount of data that we need in order to make these types of assessments. So thank you for your time.
Thanks, Jamie. I'm going to switch over and show you the Mentimeter questions that are coming through. So hopefully you can see my screen.
I think I just stopped sharing. Let me just close out my other screen here.
Okay, can you see that? And you could choose a question to answer, or I can read them out to you.
Are the GoPro percent cover determination methods available? They are covered under a quality assurance project plan. You can go to the Save the Sound website, and those QAPs, you can download them right from the website. We have both the field method, obviously, and the analysis method is also very detailed in those SOPs. I find it interesting that Chetomorpha blooms occurred in 2014 in 2 completely different regions along the East Coast. Something about global. I wonder if anywhere else. I agree. I think that the chaetomorpha blooms are really intriguing. We had seagrass in that system until the early 90s. It switched over to a mix of macroalgae by the early 2000s, and then by 2010 it was this massive Cladophora bloom, and we don't have a good reason for it. There was no change in the nutrient loading. We think it may have been temperature related, but there may also be something else going on. Caroline, if there are any that you see that I should prioritize.
Well, there are several questions about some of the methods, which is really interesting and something we could follow up maybe afterwards, getting into the details of the methods. There's one, I've read about kelp aquaculture as a nutrient removal BMP in the Long Island Sound. Is that part of what you were talking about? Is kelp as well as, these macroalgae that you're talking about?
Yeah, so we have a bioextraction coordinator in Long Island Sound, and so that person and both Connecticut Deep and New York State DEC are 2 state agencies and the Long Island Sound study are encouraging the expansion of bioextraction. Kelp is one of the ones which gets a lot of focus, as well as aquaculture. We already had our shellfish aquaculture established, but we have a number of farms, both on the Connecticut side and the New York side, that are doing kelp aquaculture in Long Island Sound.
Wow, that's really interesting, and there's a bunch more questions. We do have a panel discussion later, but if not, we'll follow up after the meeting with Jamie to make sure she can answer some more of those questions. So thank you so much. Really, really awesome talk. And so we're going to go to our next talk. So hang in there with us. Dr. Dave Demasco from Sarasota Bay Estuary Program is going to talk about using macroalgae in management in Newport Bay.
Hi, hopefully everything is showing up the way I want it to.
Yep, looks good, and we can hear you.
All right. So first off, I want to make it very clear for this presentation, I don't mean to speak for the regulators in the state of California. I don't mean to speak for the regulated community in California. I think this is a good example for our workshop about macroalgal responses to going on a nutrient diet. Did it work? But again, not speaking for the folks who regulate water quality or the folks who actually respond to that. This is, however, publicly available information. I got involved with this a couple of years ago in my former life in consulting, working for one of the regulated entities that actually seems to be part of the solution. So Newport Bay is in a beautiful part of the country, Southern California. It's in Orange County. As you might expect from the name, Orange County, there's an awful lot of agricultural land use in the area. But for the most part, this part of California, if you've ever flown in Southern California, you know that it's highly urbanized. This watershed is centered around the city of Irvine. And there are 2 main drainage features that come together. One of them is Peters Canyon Wash, which is kind of like you call them an arroyo in like New Mexico, Texas. And then Upper San Diego Creek, they come together to form San Diego Creek, which comes in not to San Diego Bay, but to Newport Bay. If you notice where we have the city of Irvine, look to the north and a little bit east, and you see Caltrans denitrification. Caltrans is their version of FDOT. It's the state transportation entity. The denitrification is a groundwater denitrification system that we're going to talk about later. And then a little bit to the west and south is IRWD McPherson Treatment Plant and Wetlands. IRWD stands for Irvine Ranch Water District, the people I worked for. They're sort of like an all-encompassing public entity that does the entire water cycle. They not only produce water through capturing in reservoirs, but they also treat the effluent that comes out. I'm not sure we have anything really like that in Florida, but the whole water cycle, not just delivering it, but then treating it afterwards, and then how it then comes down into Newport Bay and the effects of Newport Bay. So that's what it looks like, just to orient yourself. This plot right here shows, this is kind of like a display of the topography of the system. So if it kind of looks wrinkled, then that means you've got mountains. You've got the Loma Mountains, or Loma Hills, I think they are, and then the San Joaquin Hills down here. And if it's flat, that means there's houses. If you've ever flown into Southern California, you know that New York City's got nothing on the amount of development that you have in the flatter areas in Southern California. A couple of really neat features about this. Again, it's Orange County. It used to be an awful lot of citrus in this area, but not so much anymore. The citrus was occupying areas that were the flatter areas that have now been developed. And what they've left behind is a high nitrate, shallow groundwater aquifer. And by high nitrate, I mean 50, 70 milligram per liter nitrate, mostly in the form of nitrate, mostly in the form of inorganic nutrients, very high levels. But that's not where the agriculture is anymore. This is an area they used to call the frog ponds. Now the agriculture is tucked into the foothills, really, in the area that's harder to develop for residential. And that, if it's harder to develop, then it's a little bit more affordable, and that's where ag has been pushed up to. These nurseries are no longer really producing food crops so much. That's now in the Imperial Valley, farther to the east. They're producing, for the most part, crops that are used for residential landscaping, that sort of stuff. So you've got current agriculture, you've got historic agricultural influence, and then you've got residential development all throughout this area. So there is a Tmdl for macroalgae in Newport Bay, and it's based upon this. This is a photo, a couple of photos that are taken. This is part of the Tmdl, sort of like the monitoring of the implementation of the Tmdl. Is it working by researchers at Regional Water Quality Control Board? And these are photos of what it looked like in Newport Bay. Now, this is in Newport Bay itself. Now, you know that it costs a lot of money to live on the water in Florida. Imagine how much more it costs to live along the water in Southern California. These houses weren't worth, I don't know what they were worth in the 1980s, but right now these houses in this area are worth $4 to $6 million. Okay, that's kind of like a cluster of 3-bedroom, 3-bath houses, or $4 to $6 million. They're paying a lot of money to live there. They were paying a lot of money for property taxes, and this is what they were looking out at. This is the aesthetics of living along the water in Newport Bay back in the 80s and 90s. This is obviously a staged photo, but I don't know if I would send my kids out on this boat. This is a combination of identified at the time, ova, enteromorpha, and ceramium. I don't know if enteromorpha exists as a genus anymore, but it's, everyone knows what enteromorpha looks like. It's kind of tubular ova. It smelled horrible in this location, according to people, because not only did you have the macroalgal blooms themselves, and if you've been around dense macroalgae, you know it doesn't smell very good at all, but the fish kills associated. These are different types of sea bass that are common in that system. And the Band-Aid approach would be sending people out here to rake this stuff up, not to quantify it, but to put it in garbage cans and take it away somewhere. I've seen this in 2 other locations, Saudi Arabia and Sultanate of Oman. And this is not a fun job. This is not a job that actually is very efficient. It's a Band-Aid on a much bigger problem, but it shows the sort of the reason why people wanted to do something about Newport Bay. The other thing is, this is a little bit what it used to look like in Hillsborough Bay. The basis for a lot of the things we've done that have brought about improvements in Tampa Bay is this kind of phenomenon. Macroalgae along the shoreline, aesthetic impacts to high dollar real estate. That's what drove the determination to do something about the problems in Newport Bay. It persisted until the early 2000s. And actually, this is one of the examples of the environmental impact. Not only do you have the aesthetic problems, not only do you have the fish kills associated with the DO, when you use up all the oxygen in the water column, your final electron acceptors shift to sulfate, and the end product of that is hydrogen sulfide. So now you've got not only the stench of the algae, the fish kills, but the rotten egg odor of hydrogen sulfide. But this is an impact to one of the flyways, important flyway along the Pacific coast, is places like Newport Bay. And think of plovers and sandpipers, and think how they make a living by going all along the shoreline and picking stuff out of the sand. They can't do that. So we've displaced an awful lot of the habitat for important shorebirds, resident and migratory shorebirds in this particular area. So the identified impacts of this macroalgal biomass, this is the same list you could come up with in lots of different places. Displacement of waterfowl due to covering these intertidal flats with macroalgae. Loss of seagrass habitat, documented loss of seagrass habitat, not because the water got more phytoplankton, it did, but also because of the overshading associated with macroalgae growing on seagrasses. Impacts to the benthic and pelagic communities because of problems with dissolved oxygen. Macroalgae grow, it's true, but they also decompose. And when they decompose, they burn up an awful lot of oxygen in the water column. And one of the most important things is aesthetics. Never underestimate the importance of wealthy people finding their property values are being diminished because of aesthetics, smell and sight, and the fact that it's no longer an amenity to live on the water, it's now a detriment. So identified causes, a lot of really good work was done here. I want to give a particular shout out to Martha Satula with the Regional Water Quality Control Board. Used to be got her PhD at LSU. Emily Keenan knows her very well. Nitrogen limiting factor, but phosphorus at some locations and sometimes. That's important, but it's also important to note that most things that you do, not everything, but most things that you do to go after nutrient management don't act on just one nutrient. Most wastewater processes, most stormwater treatment systems act on both. It's not like we pick and choose. There are some exceptions, but for the most part, it's important to know your nutrient of greatest importance. But most systems that we put in place for dealing with wastewater and stormwater don't pick and choose which nutrient. They usually go after both, maybe with different efficiencies. They found that episodic doses and chronic enrichment were both capable of stimulating growth, which is important as we get farther in terms of what is an episodic dose, what is chronic enrichment. The major loading sources, agriculture, both current and the effects of historical urban stormwater runoff, and very important, nutrient-rich base flow, which again encompasses urban stormwater that soaks into the ground and agriculture that actually affects the water table. People who've lived out here said they didn't used to have these problems in the 60s, even though they had a lot of agriculture in the 60s. So it might be that something created the conditions that allowed the agricultural land use of prior decades to become a problem now. And that might be things like drainage that we put into the system. But the macroalgae started to become noticeable in the 70s and the 80s. We had large scale problems in this particular area. Around 1990, the Regional Water Quality Control Board, which is sort of their version of like DEP and the Water Management District rolled in one, issued permits to agricultural discharges that required sort of like recycling. You can't do that with all crops. You can't do it, for example, with lettuce, something where the water that you bring on there might actually touch something where people might eat it directly. But you can do it for like something with a rind, like oranges. You can certainly do it with plants that you grow for residential landscaping, but required where possible, where feasible, to recycle off-site movement of irrigation water to use it again. IRWD, Irvine Ranch Water District, has a large wetland, it's about 300 acres. They use it as a final polishing system for the wastewater that they process for the city of Irvine and its residents. And what they found is that it was doing such a good job, the plant and the polishing wetland behind it, that they had the capacity to do more than just their own wastewater. So they started to actually divert water out of San Diego Creek to come into this wetland to then be treated additionally. So an additional source of water to be cleaned up, if you would, through that wetland system. in the late 1990s, one of the neat things that happened is something you're not going to find in Florida, which is a groundwater dewatering process was required to have a denitrification component. So large public entities in Southern California have to deal with seismic events. And I've been in California for 2 small earthquakes. Didn't freak anyone else out, scared the hell out of me. But nonetheless, in an area with a high water table, one of the things that's noticed is that the water table acts as a lubricant, allows things to move a lot more. And so one of the things that's done by large public utilities, for example, is to lower the water table, to reduce seismic sensitivity. And that results in an offsite movement of groundwater that in the past was just discharged into the creek. Well, in the late 1990s, it was decided that that groundwater enrichment was, the groundwater enriched with nitrate was now going to have to run through a denitrification system, a large carbon wall, if you would. We'll talk about that later. So they had a very aggressive Tmdl, and that was to reach a 50% reduction in summertime loads and in wintertime loads over a short period of time. And the nice news, and we're going to talk about it, is not only did they reach it, they far exceeded their goals, and they did it earlier than people thought for some of them. And we're going to talk about the system responses. So a Tmdl based on macroalgae requires some sort of estimate of the relationship between nutrients and macroalgae, and a lot of different ways can be done. And we're going to just talk about 1, 2, 3, 4 of them, okay? One of them is a reference time period. My personal opinion, this is a good way to go in Sarasota Bay, maybe some other areas, because we know there's more macroalgae in Sarasota Bay. A lot of things have happened in Sarasota Bay, but one of the things that's happened in the last 5 to 7 years is our nitrogen concentration in the areas where we have the most macroalgae is higher on average than it was in the worst year of the 15 years before that. I don't think we have to work too hard to figure out what might be happening in parts of Sarasota Bay. We have jacked up our nitrogen concentration. We have more phytoplankton. We have more macroalgae. We have less seagrass. I think that's a good way to go. That's one way you could go. Reference location. This is kind of an approach that's being used actually in the Florida Keys, where looking at the water quality and the biological health allows you to figure out what is a water quality target that might be applicable for your system, okay? So you have reference location for NNC if you're going to do a Tmdl for a place like Biscayne Bay, where there's certainly problems with macroalgae that are ongoing right now. Empirical modeling. This is the kind of approach that when you think of the work that's been done in Tampa Bay, the wonderful data set collected by Roger Johansson and Walter Avery, just under-recognized how important their data sets were. Expanded upon by Tony Janicki and all his good work for the Tampa Bay Estuary program. And that's good where you have a lot of data, where you have over 100 sites where we're collecting chlorophyll, nitrogen on a monthly basis for 20 years. We don't really have that for macroalgae. We're not collecting macroalgae every month. We're collecting it maybe once a year, maybe twice a year. So we don't have that amount of data that can all be compared for empirical modeling. Maybe it could be done, maybe not. Mechanistic modeling is the approach they use in Newport Bay. We're going to talk about that, but a mechanistic model was the basis for predicting what will happen to macroalgae, but it wasn't the only thing they did, and that's important as well. So every mechanistic model is dependent upon a conceptual model. If you ever were a student of Clinton Dawes, you could do this in your sleep. Okay, so a lot of people, Brian LaPointe, myself, anyone who came out of Clinton Dawes' laboratory has done these kind of experiments where you look at what affects photosynthetic and respiratory rates of macroalgae. Well, there's things like temperature, salinity, water clarity, light availability, but also nutrients. We're going to focus on how important nutrients are. So this is the basis for a mechanistic model. And when you look at a conceptual model, the squares are what we call state variables, and the arrows are what we call rate coefficients. Now, there's a lot of data on those state variables. Maybe not a lot of data in a lot of locations, or maybe a lot of data in some locations, but not over the course of a year, particularly with macroalgae, where we might be sampling it once or twice. But the real data gap, if you would, are those arrows, because not many people have done work of photosynthesis respiration. Or if they have, they've not done, sediment remineralization rates, or the transformation of organic nitrogen into ammonia, that sort of stuff. But that's a conceptual model. This is a conceptual model for nitrogen dynamics that was used in this effort as well. Starts to get even more complicated because now you're starting to bring in the fact that the water mass moves around. There's an inflow of nitrate ammonia. Notice the focus on inorganic nutrients as a form of nutrients that they're particularly concerned about for macroalgae. Then you move the water in, you move it out. Again, you've got data on the things in the boxes, but there's not an awful lot of data on the rate coefficients. So the rate of uptake of macroalgae, there's a little bit of data. Brian LaPointe's probably collected more data than anyone in Florida that I know of. But we don't have data, for example, on diagenesis and the role of sediments. Remember from Roger Johansson's talk, there's a big reduction in macroalgae in Hillsborough Bay after the point source load reductions in the late 1970s and early 1980s. But it took about 10 years. It took about 3 years for phytoplankton. The role of the sediments is really important. How much data do we have? Mortality, respiration rate, so all that sort of stuff. When you put it all together, turning a conceptual model to a mechanistic one is about 90 different state variables and rate coefficients, about 90. A couple of years ago, I did a project for the Water Management District of Pinellas County on using WASP model for Lake Tarpon. And we looked at one of the more common versions of WASP, and there were about 17 different rate coefficients at a minimum that we need to know. We looked out throughout the state of Florida, talked to people like the late Carl Havens and others, and we said, well, how many of these have ever been studied? And the answer we came back with was two rate coefficients studied. So if there is an issue, going with a mechanistic model, but it is an approach that was used here. So can you model macroalgae growth? And the answer is yes. And moving forward, I'm not criticizing the people that did this. I think they did a really good job doing something they were told to do. I'm just not sure that we needed to go that approach. So here are the model estimates of what we would do and what would be the response to macroalgae associated with not only current conditions, but future scenarios moving forward. And so, yeah, we can model macroalgae growth. Notice the peak abundance is occurring in the summertime. Summertime in California is the dry season. And the dry season is driven not by stormwater, but by base flow, groundwater that manifests itself in these systems like San Diego Creek, not stormwater. Stormwater occurs in a wet season, which is their wintertime. So can you model macroalgae growth? Yes, you can. You can model anything. How good of a fit? Man, I tell you what, when you talk about physical parameters, these guys knocked it out of the park, just like most people can. If you ask someone to model water levels, salinity, water temperature, they can do a great job. And these folks did do a good job because they're very good modelers. They really are. But here's macroalgal biomass. I'm not picking on the worst. I'm not picking on the best. These are the first two graphs. The blue line represents model estimated macroalgal biomass. The red dots represent measured macroalgae. The closest fit is 25% difference. The next closest fit is 50% difference. Some of the others, it's kind of like an infinity difference because it was measured to be a lot, measured to be none, and modeled to be a lot. So would you go in an airplane where this was the fit between modeled aerodynamic capacity of the system to like lift 80 people in the air versus measured? And of course not. But we ask people to spend millions of dollars on nutrient management based upon load reductions, based upon mechanistic models where this is the fit, or something like this between what actually is measured and what is fit. So just keep that in mind as you move forward with any type of Tmdl. What goes into a mechanistic model? The clients and the consultants should be completely honest about the expectations moving forward. And a lot of times, this really good work wasn't fully vetted in terms of how comfortable we should be on, a target of 1 versus 0.5, for example. So luckily, this Tmdl had numerous goals. It wasn't simply tell me what I need to do to get my algae down to 0.5. It had concurrent water quality targets, and they're high. The DIN target was 5 milligram per liter in the upper reach of the watershed, 13 in lower reach. And you say 13, oh my God, that's way too high. groundwater in that area was 50, 70 milligrams per liter. So they're asking for an awful lot to be able to get to the load reductions that they're asking to reach. There were summer and winter nitrogen loading targets. So summer is the dry season, winter is the wet season. So what I want to talk about is what was done to get to the Tmdl, because this is, I think, the most interesting part, at least to me. The totality of the things that were done is not just a typical, here's your stormwater retrofit, ponds that you have to build. Constructed wetlands. San Joaquin Marsh does more than treat their own effluent. They actually, under no real obligation, they treat the water in San Diego Creek because they have the capacity to do it. Subsurface biofilter, La Cienega, is actually sort of like a wetland system where the nitrogen-rich water is pumped into sort of a gravel bed where the root zone is for wetlands. So it's a little bit different than a flow-through wetland, which is IRWD. Groundwater denitrification system for Caltrans. This isn't new technology. This is a concept that's been around and been actually used in Australia and New Zealand for decades. Septic tank replacement programs, good idea. And actually, but not a major focus of it because there weren't that many. Most of the places were on centralized sewage. Nursery runoff recycling programs was a very early step that they took. Stream restoration, some really cool stuff. We're going to talk about stream restoration in a minute. Not only the ability to reduce nutrient loads by reducing bankside erosion, but benefits of stream restoration that I think are really cool, that are worth looking at. And things that don't cost any government agency any money at all. Low impact development, with an area that is so heavily urbanized, reducing the individual nutrient footprint of residents in this watershed is something that actually was really promoted and something that we need to do a better job of, I believe, in Florida as well. So this is the Irvine Ranch Water District's surface wetland treatment system. It treats wastewater and it treats stormwater. I've never seen people kayak on it. I don't remember right now. I'm not sure you're supposed to kayak on it because it actually is. This is an industrial basic facility, but it's also a neighborhood amenity. A wetland system that treats wastewater and stormwater doesn't have to be a nasty little pond behind a chain link fence. It can look this pretty. Think of Sarasota County celery fields. It's a beautiful system. It's also a stormwater treatment system. This is a neighborhood amenity with pathways. People walking through here, you pay more money to live along the side of this to be able to look out over it. Most people have no idea what the system actually is doing, why it was built, and what it actually does. One of the neat things about this particular system is its very efficiency is being reduced because of everything else that's being done. So I got to work on this system because they were finding it harder and harder to meet their performance objectives. So they were expected to reduce nutrients a certain amount with a certain efficiency. And what we found is that their efficiency increased with inflow concentration. This is on a 20-year basis. And what we're finding is the nutrient concentration coming into it was decreasing. So as the nutrient concentration decreased, not only were they reducing less nutrients, they were less effective at reducing nutrients, which actually is a good problem to have. But what it means is that everyone upstream of them doing their jobs is making it less necessary for them to do what they're doing. They're still doing it. They're just not getting as much nitrogen. They're not as effective as they used to be because everyone upstream of them is doing their job as well. Again, that's a nice problem to have. Not a lot of places have that kind of problem. So,
Dave, I just wanted to see if can I give you 1 minute and then we'll move to questions or?
Yeah. Okay, stream restoration, dude, here we are. We've reduced our nitrate concentration 40% by healthier denitrification capacity. All right, in-ground denitrification, this is from Sarasota County. This is Steve's Spa system. So let's get to real quick stuff. Is it working? Here's the TIN reduction in this system. It met its TIN load reduction for the Tmdl. And what did the system respond? Algae hasn't even been noted since 2012. They reduced it dramatically. So let's just end with this. What are you going to do with your Tmdl? Well, the endpoint of the Tmdl is to actually not have a model. The endpoint is the system response. You'll never have perfect knowledge, but whoever managed with perfect knowledge. A couple of things that might make sense. when you decide upon a modeling approach, understand the weaknesses of any particular approach you do, algorithms, depth of literature, the assumptions, importance of different types of inorganic loads. And I just like to end it pretty much right there, which is we're all struggling with, do we have enough information? When did we ever have enough information to come up with management strategies? That's not what they did in Tampa in the 1970s. So don't let perfect be the enemy of good. And I'll end with that.
Thank you. That's great. I really wanted to get to a few questions. So I just wanted to jump over there real quick. And if you want to put more questions in the Mentimeter, please do so, and we can get to them today. We'll get them to Dave later. So there's three. I don't know if you can see them, Dave, but the first question is, besides the aesthetic and habitat perturbations, those anaerobic algal mats are excellent substrates for pathogen growth of public and wildlife health risk. Do you want to comment on that, or what the state of knowledge is for that?
Yeah, absolutely. I think one of the neat things about our indicator bacteria that we have, fecal coliform, enterococci, E. coli, they can come from humans, but they're not specific to animals. You can get very high levels of all three of those indicator bacteria from decomposing seaweed. So absolutely 100%. There are reasons to reduce that kind of biomass for public health reasons as well. You wouldn't want your kid to swim in water contaminated with sewage. You wouldn't want your kid to swim in water that's contaminated like an underwater compost heap either. And I think that's
second question, how has climate change and or sea level rise been a factor in macroalgal blooms?
Well, I think it, what people have talked about before, maybe climate change is making us more susceptible to algal blooms. But, the way I look at it is kind of like the opposite. We've had climate change, we have sea level rise, and we have a lot more macroalgae in some systems. We have a lot less in Newport Bay. We have a lot less macroalgae in Hillsborough Bay. And it's not because we stopped climate change or sea level rise, it's because we went on a serious nutrient management approach. So yeah, I think they're complicating. I think they could make places more susceptible. But we've actually been working in the opposite direction. We actually seem to have some success stories. And we used to be a success story in Sarasota Bay and Tampa Bay, and we can be one in the future. I think it makes it harder, but you can work against that. And Newport Bay is proof that you can work against those other stressors.
That's a great answer. And I did notice on one of the ranking questions that while climate change is a factor and we need to be monitoring and filling in some knowledge gaps there, it wasn't as high a priority as some of the other drivers. Okay, one last question before we go to break. How hard is it to model the origin and fate of floating or suspended algal drift? So how hard is it to model?
I think it's really hard. And I think that it's not necessarily necessary. We can talk, that's a topic for like an hour-long discussion, but it's really hard. And I think they did a really good job of getting as close as they could. But the reduction in macroalgae that was found in Newport Bay was not because they stopped sea level rise, not because they stopped the water from getting warm. It's because they did a serious job of reducing nutrient loads. Same thing happened in Hillsborough Bay. The same thing is happening in reverse in Sarasota Bay. Our macroalgae increase isn't occurring, without any clue as to why. Our nitrogen concentrations are higher now in the last 5 to 7 years than the worst year of the prior 15. I don't think it's by chance alone. So yeah, I think you can do it, but that kind of gets to what is your job? Is your job to have perfect knowledge about macroalgae, or is your job to come up with enough information to guide management in a way that might be expected to be, productive?
Great. Well, I'm going to stop it there. We are actually taking a break now. So it's 10:40. We're going to take a 10-minute break and be back at 10:50. And we're going to have one more talk by Chris. And then we're going to do a very short panel discussion. So see you in 10.
Hey, Carolyn, this is Chris.
Hello.
Hey, I can condense mine a little bit. I think we're a little bit behind schedule. So if you like, I can try to keep it at like 10 minutes if that would help.
It's up to you. I think if we shave off a minute or two on each, like we have you and we have the panel discussion and we have my section. So as long as we kind of keep them a little bit, let's spread it around. And so I don't want to cut you off too much.
Okay. Thanks.
Mm-hmm. Dave, you're still unmuted. I don't know if that's. Hey, everybody, we're going to get started in about a minute. All right, it's 10:50 and I want to get started. And Chris Anastasio is up with macroalgae monitoring in Kings Bay, Florida.
Thanks, Carolyn. Good after, or I guess it's still morning. So good morning, everybody. Happy Friday to you all. Thank you for giving me the opportunity to share some of the work that we've been doing in the Kings Bay Crystal River estuary, which is a little bit further north of where we are, where I am right now in Tampa Bay. I hope that this talk gives you guys a little bit of a different perspective on how we're managing macroalgae in a really different system. It is estuarine, but as you'll see in a minute, and those of you who are familiar with Kings Bay, you'll know two things. One, we don't have submarines there. We're not Kings Bay, Georgia, Kings Bay, Crystal River, Florida. And two, the drivers that really form the whole Kings Bay ecosystem are really unique. For one thing, the Kings Bay system is part of what we call the Springs Coast. So the Springs Coast is what we consider the land of mermaids and manatees, because there are a lot of both. If you've been to Weeki Wachee Springs, you know that's where the mermaids live. There are many springs in this region of the state. We have over 150 springs that we've documented across our district, our water management district, which you see outlined in that orange line, most of which are really confined up into the northern region of our district. We have 5 first magnitude springs groups, of which Kings Bay is one of them. So it's not a single spring discharging, it's multiple springs that discharge into the bay. It also is an important area, even from a seagrass perspective, because all of these springs discharge into the adjacent Gulf coastal waters via their various spring runs. And we have one of the largest seagrass meadows in the United States in that region. So it's a really ecologically rich region of the state. This is an aerial photograph of Kings Bay on the left, and those little dots indicate where we've documented our springs. So we have over 70 springs that we've documented in the Kings Bay system. And we know there are many more. There are several smaller spring vents, and there's thousands of smaller seeps that discharge into Kings Bay. And Kings Bay does have a watershed, and I'll show you an outline of that in a minute. But really, most of these systems are driven by what we call the spring sheds. And so when we talk about nutrients and we talk about nutrient loads, almost all of that nutrient load in most of these spring systems is coming from the spring shed. And you can see on the map to the right, some of these spring sheds can be rather large. Rainbow Springs, which isn't too far from Crystal River, has a very large spring shed. In fact, a fair portion of it isn't even in our water management district. It goes into the St. Johns River Water Management District. So these are relatively large underground watersheds, if you will, that we call them spring sheds. And this is a picture of the King Spring, or what you see on the photo as Tarpon Hole. Pretty large spring vent, and a very popular place to see manatees. So any of you from the west coast of Florida, if you've ever learned to dive, chances are you may have done your open water checkout here in Kings Bay. So from a vegetative standpoint, historically, Kings Bay is dominated by Vallisneria americana. So it can be called eelgrass, but that's confusing because we also call Zostera eelgrass. It can also be called tape grass. But for the purpose of this talk, I'll just call it Vallisneria so we all know what we're talking about. And Vallisneria is a really unique plant. It is a freshwater species of SAV, but it is fairly tolerant of brackish conditions. So in a bay like Kings Bay, where you're sort of right on the line between freshwater and brackish conditions, Vallisneria historically has done really well. But much like Florida and in Dave's talk with California, and certainly in Long Island Sound, we've had a lot of change in this relatively small system. Kings Bay is only 600 acres, so we're not talking about a very large water body. But we've had a lot of things happen in this bay over the last 100 to 150 years. And I think it's an important point to bring up, because we talk a lot about referencing past conditions and trying to mimic those past conditions. And I think that's really important, but it's also something we have to be careful of, because going back to the 1950s, for example, for looking at seagrass in some of our West Coast estuaries, doesn't imply that the 1950s was a time of just pristine conditions. There was a lot of stuff going on in these places in the mid 20th century. And you can see on this map, or on this graph on the left, you know, even as early as 1910, there were industrial activities happening on this bay. We had the Dixon, if you've ever seen a pencil, and it's the Dixon Ticonderoga pencil. Well, Dixon was the lumber mill that brought the wood to make the pencils. So that's where the Dixon lumber mill first came into existence in 1910, was in Kings Bay. But all throughout the last 100 plus years, there's been a lot of things that have been going on. I love this picture on the top. It's a series of photographs showing you Kings Bay. And what's really amazing about this picture is it really tells a story. In 1944, most of the watershed, most of the immediate area around Kings Bay was forested wetlands. So you had a lot of hydric hammock, a lot of really intact forested wetlands. In the 1960s, that's when all the dredging and filling really started in earnest. So lots of canals. And I love this picture. This was, I think, somewhere around 1965. You can see this giant turbidity plume coming off of this point. Because of course, back then there were no regulations. They didn't have turbidity booms and things like that. So you can see the impact on that one day when that airplane was flying over the bay of what was going on. And you can imagine, if you extrapolate that out over 15 to 20 years of development activity, that's a pretty important impact that happened to this bay. And then more recently, you can see what the bay looks like now. So lots of canals, lots of development right on the bay. In the watershed, and that's what this outline is showing you, is the surface watershed, not the spring shed, but you can see the change from 1944 to 2010 went from largely forested areas to what we would consider urban. So lots of changes. And I also want to add one quick thing to this slide, which is that over the years, you know, there have been a lot of issues in Kings Bay, and we're going to talk about the macroalgae issue here in a minute. And one way that, or there were several ideas that were brought forth over the years on how to manage the macroalgae. And one of them in the mid-1960s was a misguided idea that maybe if we dump sulfuric acid into the bay, we could kill all the hydrilla and the macroalgae. And they actually did that in 1965. They backed up tanker trucks of sulfuric acid and they dumped it in one of the small spring-fed coves. Got rid of the hydrilla, but unfortunately it got rid of everything else too. So there were a series of papers that came out shortly after that basically presented data that said, you know what, we shouldn't do this again. So what was some of the outcomes of all of this change was that we've seen a vegetative successional pattern in Kings Bay, which is perhaps not unique to Kings Bay. In the 1910s into the 20s, 30s, really all the way into the 70s, hyacinth was a real problem in Kings Bay, and people did a lot of harvesting and other chemical applications to try to control hyacinth. In the 1960s, hydrilla was introduced. Now, realize that Kings Bay, as I said earlier, sits right on the precipice between a freshwater system and a marine system. So at that time, the rainfall patterns in that part of the state were very high. It was an extremely wet period from about the 1950s to about 1970, which is when we peaked in rainfall relative to the historic average. From 1970, really up until about the last few years, we've been on a decline in rainfall. And so the bay back in those days looked a lot fresher than it does today. And then you throw sea level rise on top of that, and the bay looks much more marine than it does freshwater. But at that time, hydrilla was a huge issue. It topped off pretty much everywhere in the bay. And like I said, there were a lot of attempts, some misguided, to control that because boaters could no longer get out of the bay. They had harvesters running 24/7 throughout the bay, creating these little boat channels so people could get in and out. But around the 1990s is when we started seeing filamentous algae come into the bay. And that filamentous algae started to collect on top of these hydrilla leaves, and it started to proliferate, and eventually it smothered the hydrilla in many places. And then also coupled with that, there were several years of very severe droughts. I believe like 2010 may have been one of the driest years on record. So you have hydrilla dying back because of the smothering from the macroalgae, but you also have hydrilla dying back because the salinities are increasing. And so what had happened was over the years, the bay started to trend more towards a phytoplankton dominated bay. Now notice I haven't said anything about nutrients yet, and that's coming up, but not yet. I want to talk about our monitoring network too. So the district began monitoring SAV in the bay in 2004. Actually, it was Tom Frazier and his lab at the University of Florida that was instrumental at doing this work early on. And they published some really good papers on what the characteristics of the bay looked like, both from a SAV and a water quality standpoint. So they did some amazing work back in the day. We continue to monitor SAV in Kings Bay, and we collect both percent cover, and we also estimate biomass throughout the bay. So it was this monitoring that really allowed us to kind of understand what these successional patterns look like. And this is a graphic showing you what I just explained, which is basically from the mid-1990s to the present day. We've seen a steady decline in the amount of submerged aquatic vegetation bay-wide. Now, most of that stuff here that you see in 93, 94 was almost all hydrilla. So as we get through time, we start to see a drop off, but it's interesting because it's not only hydrilla or just aquatic macrophytes, but filamentous algae too. So everything is in decline in terms of vegetation. Well, why is that? Well, when we look at chlorophyll, we can see, just like that graphic showed you, an increase in the chlorophyll concentration in the water column. We did some work with Clint Dawes. We did some work with Bruce Cowell at USF, and they identified some of the chlorophyll in Kings Bay to be mostly diatoms. So we were seeing an increase in the amount of diatoms in Kings Bay, most of which were marine in origin. And of course, as you would expect, a reduction in Secchi disc. You could kind of see the total nitrogen line here. It's really hard to see because compared to Dave's nitrogen concentrations, we're down an order of magnitude or more below what he was showing for Newport Bay. But the fact that this is kind of a straight line, will come out here in a moment, is not insignificant. And I'll talk about that in a minute. But there's one other thing I wanted to overlay on this graph that could potentially explain part of why we're seeing this reduction in SAV. And that's our friend, the manatee. So the manatees use Kings Bay as a winter refuge. It's one of the largest winter refuges in the United States. And over the years, we've seen a near exponential rise in the number of manatees that have come in, not only in the winter, but have also stayed year round. Some of the historical work that I've seen tends to show that, manatees really weren't very common back, 100 years ago, and now they're everywhere. So the whole point of that is just that we're seeing a lot of complications other than just nutrients. So it's not a simple story like in some places. Some places it is, some places it's not. So it's a bit of a cautionary tale. We also have seen some transition even in the filamentous algae. We used to see a lot of what was then Lingbia woolii, the freshwater version of Lingbia, and now we see mostly Chaetomorpha. And even more recently, we're starting to see the Chaetomorpha dissipate. So again, there are a lot of things happening in the system. This is just a graph to show you, again, increasing salinity yields, increasing chlorophyll. So it all kind of points to a changing dynamic in Kings Bay. And so there are a lot of primary drivers, a lot of issues and drivers associated with these changes. And I don't have time to go into all of them, but what I did want to do is touch on the Tmdl. Dave talked about establishing a Tmdl based on macroalgae. Well, the same thing was done in Kings Bay. It was based on the proliferation of macroalgae, specifically Lingbia and Chaetomorpha. And so the DEP came up with some targets, nitrate targets for the springs, as well as a total nitrogen target for Kings Bay itself. And that was based on some data that was done in the laboratory. So this is an example of some of the work that was done by Stevenson et al. That really formed the basis of the nitrate Tmdl, not only in Kings Bay, but in springs everywhere in Florida. And so that was a good approach, but we know now that it's a good first step, but there's a lot more to it than just meeting a nitrogen target. And this graphic is busy, but it kind of shows you that we're seeing increasing concentrations of nitrate in most of the springs, not all of them, but most of them. Now, where you see the green bars, that's where we are currently meeting the Tmdl. The red means we're not. And this is all based on annual geometric means. The interesting thing to point out here is that the graphic on the top left is the total nitrogen concentration for the bay itself. And you can see that really since 2010, we've been pretty well below the Tmdl. So we're meeting the Tmdl for nitrogen in Kings Bay itself. Not in all of the springs, but even in the springs themselves, there are some that we are meeting the Tmdl. So the Tmdl would suggest that we are below the threshold that should create the conditions that are such that we shouldn't have macroalgae as an issue, but we still do. So it's not to say that the work that was done to develop the Tmdl was bad. It was really good work. It's just that we need to constantly be looking at this and saying, are these targets appropriate? Perhaps they should be lower. Perhaps it's something other than nitrogen that's driving this distribution. So there's a lot of these questions that we've been asking, and part of that process was to create what we call the Springs Coast Steering Committee. And this is for the entire Springs Coast. So that whole region I showed you on the map earlier, and it was based on the estuary program model. So we've had this Springs Coast Steering Committee since about 2014, and it's been really instrumental in getting folks together and talking about these issues and doing things like we're doing today. There have been a lot of projects that have been put in the ground, not necessarily for nutrients, but perhaps as beneficiaries of nutrient reduction. One example of that is a reclaimed water project that was completed a few years ago, where reclaimed water from the city of Crystal River is sent to the Duke Energy power complex. And that does two things. One, it reduces the need for Duke Energy to use potable groundwater, but it also takes a nutrient source and moves it out of the watershed. Their process is internal. So the water really stays inside and there's no direct discharge into the Gulf of Mexico. So that was an example of a project that was done to help that process. We've done treatment wetland projects. This is at Three Sister Springs. This was a treatment wetland project to help treat the water going into Kings Bay. And then I wanted to leave you with one last project, which really speaks, I think, to what we heard in Long Island Sound, which is volunteers. Betty Stogler's talked about that as well. It's really important to have the community engaged. And so there's a group that started out doing a project called One Rake at a Time. And it was a volunteer effort. They would go out and they would literally rake algae and put it in garbage cans, much like the picture that Dave showed of the folks raking macroalgae. It is not a very efficient process, but what it did is it raised awareness of the fact that this is an issue. A few years ago, Save Crystal River is a grassroots organization. It's a community-based organization, decided that they were going to step that up. And they would go out, and we worked with them on a technical level to help direct some of the work that they were doing. But their focus was, we don't want to just take the algae and remove it. We want to put something back. So they recognize that, nature abhors a vacuum. So you can remove the algae, but you have to put something back in its place. So they started a really ambitious effort to actively replace the macroalgae and the top organic layer of accumulated sediment, mostly in the canals of Kings Bay that had never been maintained, with Vallisneria. And they've had some pretty good success. I wouldn't say that it was successful everywhere, but I think just the fact that they're doing this is a really important step in the right direction. And what it shows is really the idea that you have to diversify your management actions. Nutrient reduction is very important and needs to continue. But like Dave said, We don't want to get hung up on finding the exact number that we need to get to. It's common sense. We know we need to reduce our nutrient load, so we're going to do that. But that may not be enough. There was a really great body of literature that was produced in Silver Springs and Silver River by the University of Florida. It was funded and managed by the St. Johns River Water Management District, called the CRISPS study. It was a multi-year study. It was an amazing body of literature. But one of the overarching conclusions that they made with respect to not only Silver River, but all the spring systems in Florida is nutrient reduction alone likely will not get you back to where you want to be. So it's a really important thing. I'm not saying that's true for Sarasota Bay or Tampa Bay or any of the systems that we're talking about today, but it is a cautionary note. We have to think outside the box. We have to think holistically. And I think it's really important that we diversify the management actions. We don't just focus too much on one thing, but that we look at all the different ways that we can improve these systems. And the last thing I wanted to say is that there's a bit of acceptance that goes into this as well. With Kings Bay, we've seen changes, and perhaps some of these changes are related to sea level rise. Very flat place. Average, elevation in the city of Crystal River is less than 3 feet. So, 6-inch rise in sea level can have a huge impact. It also has an impact on the amount of groundwater that's coming out of our springs. We can't necessarily control that at our level. So there is an acceptance that some of these ecological changes may be permanent, but that doesn't mean we can't work with that and be the best that we can be in the system we are. Kings Bay may never be a freshwater spring again, but man, it can be a beautiful estuarine embayment. And with that, if we have time, I'll take some questions. Thank you.
Yes, thank you, Chris. That's great. I'm going to share my screen so that you can see the Mentimeter questions. One of the questions is to show your driver's slide again. So if you could click back at some point, we'll go over that. But here are the questions. One is, I'm glad you mentioned the manatees because the top-down effects are often ignored and the focus is all on nutrients. Do you think herbivores or the lack of herbivores is an issue elsewhere?
Yes.
And you just touched on that in your last comment, so.
Yeah, and I do think it's an issue. Well, I didn't really talk about other herbivory issues because of the time, but one thing we noticed with filamentous algae, specifically the Lingbia woolii, is that you disrupt the algal grazers that, we see a definite difference in grazer assemblage when you go into a Valsinaria bed versus a filamentous algae bed, and it's quite limited in what grazers there are. And so we think that there is an issue with that. Now, whether or not we can actually reintroduce grazers that have been lost because you don't have the Valsinaria beds anymore, we've talked about that, but we're not sure how to do that. But yes, I think herbivory is a really big issue. It is often overlooked. I agree.
Great. And the other question is, do you think your Tmdl nitrate concentrations are now acceptable because the high chlorophyll levels are removing or taking up a lot of the nitrate? I don't know if you can see that question or if you want to reread it.
Yeah, no, that's a really good question. It's the issue that I think we tend to focus so heavily on the water column that we lose sight of some of the nutrient pathways towards macroalgae. And I think that's the case in other systems too. I think perhaps Sarasota Bay, we see the same thing, where there are areas that would suggest, based on the numeric nutrient criteria, that your bay is doing really well. But I think what we fail to see is that those nutrient pathways, if you're not looking for that response, you may be missing it. So in Kings Bay, the Chaetomorpha people don't like because it's not Valsinaria, but it does a really good job of cleaning the water column. In fact, you can go online and buy it for your aquarium. It's a preferred type of algae that helps clean the water in your aquarium. So if you have a lot of Chaetomorpha in the bay, it may not be ideal, but it is helping to prevent the phytoplankton from, really taking those nutrients and becoming a problem in that way. I think what we're seeing in recent years, which I didn't really show in the slides, but if we're seeing a reduction in chaetomorpha, as would be the trend overall in the bay, I think what I would argue is that perhaps your nitrate or your TN in the bay needs to be even lower, because obviously your chlorophyll or your phytoplankton are utilizing that nitrogen, and we're seeing increases in the chlorophyll concentration in the bay.
Okay, thanks. I'm gonna stop sharing, and if you want to reshare and show your drivers slide, we're going to move over to once Chris shows his slide, we're going to move over to the panel discussion. And so on the Mentimeter, you should be able to have a new slide that says questions for panel. And I think Darcy might have a few follow-up questions from Dave's talk as well to put in there. So I don't know if we want to start off with one of those questions. Let's have Chris show his slide first. So there was a request to see this slide, which looks good. So maybe you want to talk just a few minutes, a minute about it.
Yeah, I mean, this slide kind of tells the story about why it's important to think holistically, and this whole idea of diversifying your portfolio is important, because there are several issues that go beyond simply that nitrogen macroalgae relationship. And one I didn't touch on, which is on the driver's list, is recreation. I just, I didn't talk about it, not on purpose. I just didn't mention it. But recreation is a really important driver in this system, because if you've ever gone to see the manatees in Kings Bay, there are anywhere between 12 and 24 dive boats out there carting people around, and they literally chase the manatees all around the bay. And then when they corral them, they dump the people in, and most of them can't swim, so they have noodles. So you get these German tourists that come to see the manatees, and they've never been in water before, so they give them these noodles. So we call them noodle people. And the noodle people, you'll just see just a sea of those pool noodles around, flailing around, kicking their hands. And so not only is that that disrupts the fauna, but it's a very shallow bay. So most of the bay is less than 10 feet deep. So you've got people with fins kicking up all this stuff. And so there's a lot of this stuff gets resuspended. And in fact, if you look at the driver of clarity in Kings Bay, it's about 50-50 between total suspended solids and chlorophyll. Now, most of the TSS is volatile, so there's a lot of autocorrelation between chlorophyll and suspended solids. But it doesn't surprise me that that's the case, because you have a lot of that fine unconsolidated material on the bottom, and you get people that are flailing about. So recreation is a major impact in Kings Bay.
Well, thank you. Thanks. So, and that's a good, we're talking and we're discussing, and I can see that the panelists have their videos on. So hopefully everyone's seeing my screen, which is showing that there's 2 questions up there. So I don't know if the panelists want to take them in turn. You can feel free to jump in. And I will mention that it's 11:18, and we're going to have the panel go to 11:30 at the least. So we'll see how we go by 11:30.
I could handle the nutrient management strategy one, since it seems to be, maybe something Jamie can chip in as well, because it seems like they're saying, other than in Florida. I'd like to build on one thing that Chris said, which is, the importance of getting out in the field. So much of our work is done by folks who are just looking at screens or models. And I remember working with Chris back in our days in the Water Management District, where we jumped into Salt Springs and Alexander Springs. These are both systems where the recharge area is Ocala National Forest. It is meeting water quality targets. And guess what? It was full of Lingbia, or whatever it's called right now. But there were people stomping around, and there was no native vegetation. And so, sometimes it's really important to understand a system, to get out from behind your desk, stop experiencing the world through Google Earth, get out in the water and poke your head underneath the waves and see what you see. And it's very different. So just a little thing, you know, make sure you spend enough time in the water to see. And that was really true for Charlotte Harbor. You know, in my prior life, I wrote the Charlotte Harbor Swim Plan. Thanks to the comments from Betty Stogler and Dave Blewett and others, that thing was stopped in its tracks because macroalgae was becoming really abundant in places, and it never was showing up in a monitoring program because guess what? A monitoring program only captures water in the water column. It doesn't capture things that aren't in the water column. So spend time out in the field, otherwise you really don't know your system that well. That would be one thing. Nutrient management strategies, I think one of the important things is make sure you're looking at inorganic loads as well as total loads. It doesn't mean ignore total loads, but particulate nitrogen is not as problematic as organic nitrogen. Organic nitrogen is not as problematic as inorganic nitrogen. Think about, you know, put yourself in the mindset of a phytoplankton cell or ulva. You're going to be able to use inorganic nitrogen immediately. You're going to be able to take it up. If it's organic nitrogen, it might have to be processed before you can even access it. All right. And if you're in a water mass that doesn't turn over very much at all, like Little Sarasota Bay or a lake, maybe you have enough time for that nitrogen to be processed. But if you're in a place that flushes, you know, 90% water exchange in 10 days, like much of Sarasota Bay, then the inorganic nutrient loads are probably the most important nutrient loads. And I think that's an important thing for us to pay attention to. Look at Roger Johansson's early work, and he looks at inorganic nutrient loads. All right, so he knows inorganic nutrient loads were a better explanation of the variability in chlorophyll than total nitrogen. And so I think that's an important thing for us to look at. So where are your inorganic nutrient loads coming from? Could be very different than total loads. Because think of like base flow and groundwater. You're talking about a mass of water that's already been filtered. And most of the nutrients coming in through base flow, groundwater, are going to be inorganic because they're kind of like run through a coffee filter, if you would. So doesn't mean you don't pay attention to stormwater, doesn't mean you don't pay attention to total nitrogen, but it means you go beyond that. Just because you have to do it doesn't mean you have to stop there. So go beyond the basics and look at what matters the most. What are the forms of nitrogen that matter the most? They're probably going to be inorganic nutrients in high concentrations rather than organic or particulate in a low concentration. So think like phytoplankton, and maybe you can come up with solutions that might be a little bit different than the traditional, hey, let's build another stormwater pond at the end of the road.
Can I go back real quick to the question on the community-based monitoring? And I was curious what Jamie's thoughts were on this as well. So one of the things that, you know, and I work with Betty, you know, quite a bit, and I think the work she's done with the Eyes on Seagrass is awesome. One of the issues we ran into in Kings Bay was, I mentioned the one rake at a time program, and it was really successful, and people came out on the weekends and they really felt good about, you know, helping their community and helping clean up all this algae. And that was, I think that was their tagline was, we're cleaning up the bay one rake at a time. But, you know, that to what Dave said earlier in his talk about, it's an ineffective way of managing the algae problem. We have to be careful about that, because what I noticed is that people were kind of getting a little bit depressed about it, you know, and they felt like all this work they're doing didn't really amount to much. And if anything, you know, they may be spreading more algae because it's a messy process. And so all the filaments go everywhere. So I'm just curious, like, how we manage that? It's really important. So I think community involvement's important, but we also have to keep, you know, we have to keep it in perspective. There has to be a bit of a reality check that, look, you know, most of these issues aren't going to go away, you know, in a month, and people want quick results. And when they don't get them, then they get a little bit, you know, disengaged about the process. I mean, look at Tampa Bay. It took Tampa Bay, what, 30 years to see the recovery that we've seen in seagrass. And it takes a long time. So I just wanted to point that out.
So I'll follow up on 2 points here. Regarding removing the algae from systems, the mechanical harvesting of algae, one of the problems that we run into in Long Island Sound is that right now we can't touch the bottom. Like we literally, even in a really heavily impacted system, we have to go through our state agencies and Army Corps of Engineers, and are likely to be shut down if we say we want to trawl or we want to rake from the bottom. So unlike in lake systems where you have these harvesters and they can pull up algae from the bottom through the movement, we're not yet allowed to do that. So it would be great if we could, but right now we are relying on people going out and removing it from beaches, or towns removing it from beaches. So there is that regulatory issue. The community based monitoring that we do is really effective in raising awareness of the issue, I think, with the groups that are doing it. And then the report card also really helps to get that word out that seaweed is a problem, because, you know, going back just 10 years in Little Narragansett Bay, where we have that 3P algae bloom, the water quality looks amazing. It's low turbidity, low chlorophyll, and that's because the seaweed is such a great filter of nutrients and a sediment trap. So everyone thought, oh, it's looking great. Why'd we lose seagrass? Well, because we have a foot of algae on the bottom. One thing I will mention about the rake toss, we just put out the report card for the embayments last year using data from 2018 and 2019. So this is a very new thing for us. When we initiated the Unified Water Study, we didn't think we were going to be able to use those rake tosses in our grading approach. We thought that the data just wouldn't be consistent enough. We were worried about, you know, well, we're targeting bad spots. What can we really say about that? What if they're all bad? You know, when we looked at the data from 2018 and 2019, we were really impressed with the ability to see patterns, and those patterns were linked to what we understood about the nitrogen loads to these systems and the flushing times of these systems. So those rake tosses, even though they are super low tech, and we're just doing none, some, lots, seem to carry a lot of information. And that's really what our goal was do something that is quick and easy and tells us a lot about the system. And that is something that, I mean, right now it's rolled into our Unified Water Study, so it's part of a larger monitoring program. But rake tosses, I think, are a really effective way to engage volunteers. And you can do them from docks, from boats, from all sorts of settings. You can get a lot of information relatively quickly and easily. So they are a great first step in trying to assess the scale of your problem.
Jamie, I had a quick follow-up. So do the people in that region, did they see the macroalgae, the seaweed, as necessarily bad? Because if it's viewed as a way to clean the water column, and so you have nice clear water, what's the perspective of folks? I mean, is it something that people are like, we got to get rid of this at all costs, or no, it's not so bad, we like it?
So it really depends on the system. The Little Narragansett Bay, nobody cared until it started washing up on the shorelines of a very wealthy neighborhood where people had the money and the political connections to really complain about it, honestly. And then it started making the papers, and then it became, you know, where is this coming from? They actually blamed seagrass, which was a little scary because the cladophora is like Velcro and captures all of this old seagrass. And so you look at it and the cladophora rots away and you're left with just old seagrass bits. And so they thought, oh, seagrass is the problem. I'm like, no. But in other areas, I think that we recognize that in our systems, the seaweed itself tends to be a problem. We want a diverse assemblage of macroalgae. And I think that may be what you're asking about. So we try to make the distinction when we're talking to the public. that seaweed is not bad. We want a diverse assemblage. We want to see reds and greens and browns, and we want to see some of it, but not a foot thick of just one type. So we try to draw that distinction between macroalgae communities are good, especially our rockweeds and our kelps, which act a lot like seagrass in our systems. But when you get just one type and it's everywhere, that's when we've turned the point and are into the bad area.
These are all really, I'm loving this discussion, and there's a bunch of other questions on here. I've turned it into a kind of scrolling grid. So I didn't know if we wanted to, there's some questions about nutrient pathways and how they affect the idea that, like, as seagrass dies, it releases nutrients. Here's one, let's see if I can pause. One aspect of loss of seagrass is the loss of stored nutrients. So how do we take that into consideration when we're thinking about TMDLs and nutrient pathways and how that affects the TMDL in the future?
I'd like to take a stab at that one, and that's actually a phenomenon we think is happening in the upper part of Sarasota Bay, and that is important. I think to understand enough about your system so that you don't go with the most simplistic without testing it. We need better data on macroalgae, but we don't need better data on every aspect to move in ways that make common sense. But we do think that part of the problem that we see in the upper part of the bay is not necessarily that anthropogenic activities caused the seagrass loss, but anthropogenic activities could cause a natural phenomenon to be worse. And part of it is, like, think back, do we have a pristine watershed? No, we don't. We don't have, we look at all the changes that took place in Kings Bay. Look at all the changes we've made to, like, the circulation in a place like Donna Bay, which now has 5 times the watershed that it had historically. Naples Bay has a watershed 10 times as big as it used to be historically. Those are things that need to be acted on. It's not just one thing that needs to be acted on. But once we have that die-off that we think occurred due to red tide, we're not so simplistic as to say red tides are caused by humans, but we're not so simplistic as to say humans have nothing to do with red tide. We're careful in saying we didn't cause it, well, we can cause it to be worse. So we are making a connection there, but we've lost a lot of seagrass. It's going to be well in excess of 1,000 acres, maybe 2,000 up in that area. And that is going to fundamentally change that system until we get the seagrass meadows back. That's an awful lot of nitrogen and phosphorus that's now available for something else. Nature abhors a vacuum. It's going to go somewhere. Hopefully, we can get that back, but it does mean, pay attention to the kind of stuff that Chris has been talking about, but also keep in the back of your mind, what is the nutrient load increase compared to when nobody lived here? If the only thing you did was modify the shoreline, okay, maybe you have to look at that. If the only thing we did was increase, manatee populations, maybe, kind of like don't do much else. But what is your nutrient load compared to what it was historically? And I don't know anywhere in Florida where we don't have more of a nutrient load than we had historically. So it's always going to remain something that makes sense for you to address. You're never going to have perfect knowledge. You can't act on everything, but you can act to do a better job with nutrients. And it doesn't always cost money. Public participation, Jamie's been talking about public participation and quantifying. Absolutely. Well, let's utilize that as well and get things like reroute the spout. There's no reason for us to have raised median strips on our roadways. Every median strip in our roadway in Florida could be a dry detention system of some sort. We can do dispersed stormwater treatment all throughout our watershed, and it won't cost nearly as much money as expecting the magic stormwater pond at the bottom of the hill to fix all our problems. So, I think it's all up to us. It's kind of like that pogo strip from a couple of decades ago. We have met the problem. It's us. We're creating this problem. And I'd like to just point out one last thing. We're creating a generation of kids that won't know what it's like to experience a healthy northern Indian River Lagoon. My daughter grew up playing in the beautiful Sarasota Bay, Tampa Bay, Gulf of Mexico. She's in college. She comes back to do spring break. If your kid actually was raised in what the Indian River Lagoon looks like right now, do you think they want to come back and experience that? It's just, Biscayne Bay, you know, we're really cheating people out of the experience that we've had. A beautiful system falling apart is cheating younger generations from something we were fortunate enough to experience. It's just not right.
Dave, I'm
Go
sorry.
ahead, Chris.
No, you just hit on a really important point that I experienced firsthand with working in the spring systems, is that, you know, those systems have been impacted for years, decades, and a lot of the folks that remember those systems back in the 40s and 50s, they're not here anymore, and so your baseline has changed because people go today. You know, you get people going to Kings Bay today, and they don't understand the problem. They're like, this place is beautiful. Yeah, because, you know, they maybe they came from, like, you know, some landlocked, depressed town where they've never seen a spring before, and they come to Kings Bay and say, man, this is like beautiful. I don't get it. What's the problem? And that's a big issue. I don't think we have that to worry about that so much in some of the estuaries we've talked about, because the impact is relatively well, it's a large impact. So, like, you know, Charlotte Harbor, take the east wall. I mean, we've seen incredible changes there, and I don't think you have to, you know, be from somewhere in Kansas to know that, you know, to recognize, oh yeah, there's a problem here. Yeah, it's still beautiful, but this doesn't look right. So, but it is a consideration that we have to take. And that goes back to my point that, you know, there are some things that we can change and some things that we just can't. I don't know how many thousands of miles of canals we have in Charlotte Harbor, but are we going to fill those canals? No. If the problem is that we have watersheds that have an intense high density development or lots of fertilizer, are we going to move them out of the watershed? No. So we have to find ways to improve and reduce our nutrient impact and go on that nutrient diet that they've talked about. But there are other factors like climate change. I mean, we may be in a different state today than we were 20 years ago. And so some of it, we have to accept that and work with what we've got and improve what we've got to the best of our ability.
Thanks. And I think one of the things I was going to say, and I do think we need to wrap up and move on to our next section, but I think it would be really cool to challenge everybody at the workshop and all of our speakers. There's been several questions about how do we come together to collaborate and coordinate our monitoring efforts so that we can fill these data gaps and we can make these, you know, change the actions that we're taking and have more success stories in the future. So I see them all kind of connecting with each other, where we're doing it. That's what this workshop is about, right? You know, everyone's coming together to share knowledge and figure out where those data gaps are so that we then can address them with management actions and hopefully have an improved system in the future. So I don't know if each of the panelists want to kind of give a 1-minute summary of what you know, Dave was just talking about. Do we upgrade our you know, our wastewater treatment plants? Do we stop people from, you know, maybe Chris, stop people from going out and viewing the manatees, and that would help, you know. So if you guys want to each give maybe one critical action that you think needs to be taken so that we can have more success stories.
Who wants to go first?
I think, you know, I can start, and that is Sarasota Bay, I think, has one of the most simplistic systems that you've heard about the last couple of days. And the reason why I think we can actually be confident that we have a pretty good idea what's going on is because we used to be a success story, and we will be in the future as well. But our change in our water quality is so evident. You know, the seagrass loss, the phytoplankton increase, the macroalgae increase is occurring over the same time period where we've had a nutrient level sustained that we didn't even have in our worst years, the 15 years prior to that. I think that's, you know, kind of smoking and lung cancer. You know, you don't have to have every last detail to know what you need to do. And I think that that's, you know, we're going to be tested over the next couple of years about whether or not we can pull it off again. Because if we can't pull it off, then I'm not sure you have a lot of luck with places that are more complicated. Our system and our impacts are better understood than a lot of places, and it's not perfect knowledge. But our solution is not the only thing that you have to do in other places that are so messed up, Naples Bay, Biscayne Bay, etc. But if we can show positive response in the next couple of years, I think it should give, you know, comfort to other places. And I do think, don't let perfect be the enemy of good. Do the things that make sense. There's no reason why we can't have AWT wastewater treatment plants throughout Florida. It's not new technology. It was new technology in the 1970s. And so money is the limiting factor. Well, guess what? Your budgets reflect your priorities. If we can find money to build roads that no one seemingly asked for, to go to places that were surprised by the fact that there's a new toll road going to Georgia, we can find money to upgrade wastewater treatment plants to do more places. There's things you can do that don't cost money. Miami-Dade County didn't have a wet season fertilizer ban until I think last year. We've had that for a very long period of time. So not everything costs public money. Not everything is like, oh, I don't know what to do about it. We know how to do a better job with wastewater. The East Coast of Florida doesn't have the same grizzle-fig criteria that we have. Why not? You know, we have to be more careful about reclaimed water. Doesn't mean you don't use reclaimed water, it just means be careful about it. 2 pounds per 1,000 square feet per year of lawn. We need to make sure that we're not adding 1,200 pounds of nitrogen.
I'm gonna, it sounds like Dave's got a prioritized list of things that we can do to make change. So I appreciate that, Dave. Maybe we can have Jamie give her very quick one word answer maybe, and then I'm sorry to cut us all off. This is an excellent discussion.
Wastewater, big issue. That's my one word.
Okay, thank you so much. Thank you to all of our presenters and panelists together. All of you guys have been so great, and I feel like I love all the questions coming in. So I really appreciate all the participants too, hanging in there, sending your questions. If your questions weren't answered, believe me, we're writing this all down and addressing it, and that is what we want to talk about next. But I have a very quick, we're going to switch a little bit quickly to a new Mentimeter. So I'm going to share my screen with a Mentimeter question, but you have to put in a new code. So if you go to a new browser, put in menti.com and type in this code 1208-9725. We have a question from Tampa Bay. This is for Tampa Bay. Please rate these suggested monitoring and research gaps for Tampa Bay. Okay, I think we're getting a lot of answers in. Really appreciate that. Seeing some folks really think habitat implications and successional cycles are important for monitoring and research, more so than maybe the relationships and sediment trapping implications. So keep putting in your answers. I'm going to go back to the other Menti presentation. Keep putting in your answers here. It should stay open. And I'm going to switch back over. So right now we're going to really go through what our next steps are and how we're going to kind of try to synthesize all of this amazing stuff into a workshop report. So I'm going to resume share. Are you guys seeing the 4 parts of storytelling? Okay, so I'm going to lead us through basically a storytelling synthesis exercise so that we can try to figure out how we're going to put all the amazing information that's been coming out over the last 3 days of the workshop into a workshop report. So I wanted to at least start with a backgrounder on what I mean by storytelling. And I've boiled it down to 4 parts of storytelling. So storytelling is synthesis, and synthesis is storytelling. So we're going to get away from summarizing results or just listing out bullet points of what we know and what we don't know, but really we need to think about how can we tell a story that tells the overall picture of macroalgae blooms in Florida, in the different estuaries themselves, but also overall. Data visualization, which encompasses everything from images to maps to figures, conceptual diagrams, infographics. Those kind of data visualizations are, including the proper design of those data, is a key. So we're going to really focus on some key visual images that we want to put into the report. Storytelling usually can be broken down into 3 kind of parts. So you have causality, which is what's happening, what's causing these problems. Temporality, so time frames, usually over some period of time. It could be, time travel. So you're, jumping between historic and current. And the characters involved. So what is the character? What's the main character of your story? I mean, AKA it's going to be macroalgae, but that character can change, and there's always sub-characters. There's also tools that we can use to really get at storytelling. So it's not just an empty void that we're trying to get you guys to fill. Tools such as using active titles, and but therefore statements. That's a Randy Olson exercise where you really try to combine the different parts of your story with here's what's happening, here's the tension, and here's the resolution. A very common use of storytelling is the hero's journey. So look up the hero's journey and find out more about that. And we could maybe set this up to be a hero's journey type story. So after watching, listening, and taking in all the information from the workshop, I have come up with what my thoughts are on the report content, but using active titles. So active titles are something that we use fairly frequently to summarize key points and tell the reader your answer. And so they don't have to read through paragraphs of text. They're just getting from the heading. If all they read is the heading, they know what the answer is. So some of my active titles that will then help you set up a table of contents or contents within your report. I have macroalgae are diverse, not well understood, and increasing in Florida estuaries. That could be almost like an intro section. The drivers of macroalgae blooms are nutrient pollution and changing hydrology. These are obviously very simple statements, and they will have a lot of supporting information, but trying to get at those high levels as we're listening to these presentations over the last 3 days. Significant data and knowledge gaps are on macroalgae ecology are hindering our understanding of these estuaries. Ongoing research and monitoring of macroalgae and related studies is needed. So these are kind of examples of active titles, and I would encourage you to take these, edit them, respond to these questions, figure out what's working, what's not. And again, these are kind of my suggested thoughts based on the presentations. I also thought there were several other different parts of the content that we really need to think about. So, we have a question, what are we going to do about Caulerpa and Limbia? It seems like Caulerpa came up here and Limbia came up here, and it doesn't really fit into this box that we wanted to fit in. So I thought that would be a really perfect place for like a breakout box or a special story about that, particularly. Lessons learned from all the estuaries, obviously. Historical and current perspectives of macroalgae, and this is kind of getting at that storytelling. So it would be really great if we could have people tell a story and highlight that story in the report. So, from your personal perspective, I used to go down to my dock and I would have clear water and seagrasses on the bottom, and now I have nothing but phytoplankton, and I can't even see if there's macroalgae there or not, you know, and it would be a quote from someone. So I think that would be a really great way to incorporate historical and current perspectives, but from a people point of view. Citizen science monitoring can fill gaps in knowledge. That's a good, excellent active title. And I think it would be a similar, like a breakout box highlighting the citizen science monitoring that's already occurring, and almost like a call to action to get more people involved in community-based science. Applying management success stories from other regions. So this was obviously from today. So it would be really great to include a hopeful note of how we can help improve the system, and using, you know, lessons learned from other systems and applying them to these estuaries. And of course, we always want to have recommendations and next steps in a report. So this kind of is my idea of the content for this report, and I would love to hear people's feedback on these. So I'm just going to pause there. Let me go back to this. I can't see you guys, which is a shame. But if you could write into the chat, I know that there's some people already chatting, but if you would like to write into the chat some of your either changes to these active titles or content for the report, I would really appreciate it. I'm just going to pause for about 1 minute and think about what you like, what you don't like. Don't worry, I have a question about what isn't here in a little bit, but let's just pause there for a minute. I see some hearts are coming through, so hopefully that means that people think I've kind of covered the bases so far. Great, and I'm going to keep going. So unfortunately, I don't think we have time to walk through a storyboarding activity together. There's a Jamboard available that I was going to do some mock-ups of the different pages. So that's a fun interactive way to get some feedback from you guys, but I'm going to skip that for now, and I'm going to go to a Menti question for you guys. So what is a report if not without a really great title that people are going to want to read? So this is an open-ended question for everyone for your suggestions for the title of the workshop report. Thinking back to, you can put it in the Menti. The Menti code is the original Menti code of 74461202. And I would love to see some answers to this. It could just be as simple as, you know, Florida's macroalgae workshop report, or maybe it'll be a little bit more active. Nice. That's great. These are really good. I love it. These are really great. I can't seem to scroll. Let's see if I can get. There we go. Okay. We're trying to make it scroll. There we go. I do like all of this. I think this is great. The creature from the Indian River Lagoon, of course. So that's really, really great. Florida water or what's underneath. These are really good. I'm going to take these. I think there's some really good ones here, and maybe we can do some kind of ranking Menti later on. But these are all really great. Keep going. Everybody's getting the hang of this. All right. So if we go on, let's see. Okay. One of the things that always seems to crop up in these types of reports is you've got the front cover, which we just talked about, and the title that's really going to jazz it up. On the back side, in case no one ever cracks open this report, you've got the front and the back. On the back, I'm asking you to rank what you think absolutely should be on the back cover. The back cover is actually pretty good real estate. And so there's lots of stuff we could put in there, but I'm asking everyone to rank what they think is the most important part if someone doesn't even open the report. Great, everybody is really doing a good job with putting in their answers, ranking everything. All right, people are still answering, so I don't want to move on quite yet. I do have I did do a storyboarding mock-up of the report, and it seems that a lot of the answers that you guys have are already in that, what I thought was a good way forward. So I look forward to working on that more after this. I'm going to take the information from these Menti slides and what you liked and what you didn't like, and help to formulate a synthesis and a story from this workshop. So I really appreciate that. I'm just going to go on to the next slide. I really appreciate that people are still putting in their answers. What was one point that should be included in the report, and was it missing from our outline? So there is one thing that you feel like has to be put in this report, and especially if it wasn't already listed in the content slide, what would it be? Oh, am I sharing? Can you guys see that now? Policy recommendations. While people are doing that, I'll go back to the actual content slide so you can see it. Whoa, too far. There we go. This is a quick reminder. Here are the contents. Okay, so going back to what is that one thing that needs to be included and that probably wasn't on my outline. We've still got policy recommendations as the biggest. Monitoring goals, that's a good one. Really stating what our goal is for each of these monitoring programs, or as a whole, a consensus on what are our goals for monitoring macroalgae. All right, there's lots of individual stuff, but I'm sure there's stuff that's going to overlap. So I see a lot of management, thinking about economics is in there. You know, so it does come down to management, regulation, policy. Yeah, the importance of monitoring. All right. Well, I really appreciate that. Keep going. If you haven't filled it out yet, I'm going to go to the next slide. Well, I think that we've probably covered our 4 parts of storytelling, so I just wanted to touch base. We've talked about data visualization. We didn't get to the storytelling, or the storyboarding, but I do have some ideas about that. Causality, I think we're fairly clear about the cause of macroalgae blooms, but we're going to talk a lot about that. We've been talking a lot about the historical perspective, current and historic status of these bays and estuaries, and the characters and the subcharacters. And we've used active titles and more. The Menti is a great way to get feedback about what we should include in the report. So I just want to say very quickly a thank you for everybody for participating, and also for the speakers and the steering committee. And I wrote a quick haiku for you. Macroalgae bloom, murky, gooey bays result. Teamwork to fix it. So thank you. I'm going to hand it over to Darcy.
Thank you, Caroline. It kind of hurts not to just end on that awesome haiku, but I do have a few more people to thank. I know acknowledgements fell to the bottom of our priority list for that back page, but we do have a lot of people to acknowledge who helped pull this workshop together. First of which is all of you for participating and taking 3 mornings this week to be here and sharing your insights with us, especially those of you who attended those breakout sessions on Wednesday. Thank you so much. And we will be following up with you via email with some follow-ups, ways that you can keep in touch with us, ways that you can see those speaker presentations. I know a lot of you would like to follow up on those afterwards, as well as the meeting recordings once we've been able to clean those up and get them available. So please watch your emails. I want to thank all of our speakers Monday, Wednesday, and Friday for sharing their expertise and participating on those panels. I thought today's panel discussion especially was just fantastic. Thank you to Caroline for being a fantastic workshop facilitator, taking us through this storyboarding activity, which will help us put together a really interactive and responsive workshop report that we'll be getting out to all of you. And thanks also to our facilitators on the Wednesday breakout groups for taking our geographic areas through those questions. And then finally, I want to thank our steering committee members by name today. We had on our steering committee working with us for the past number of months to bring together all of our speakers and participants. Nicole Idavia and Jennifer Hecker with the Coastal and Heartland National Estuary Partnership, Gary Rollerson and Ed Sherwood with the Tampa Bay Estuary Program, Jay Leveroni and Dave Demasco with Sarasota Bay Estuary Program, Chris Anastasiou with the Southwest Florida Water Management District, Chuck Jacoby with the St. Johns River Water Management District, Dennis Hanasak with Harbor Branch Oceanographic Institute, and Betty Stogler and Armando Ubeda with the Florida Sea Grant Extension offices. So thank you so much to everyone. I really hope that you got a lot out of this workshop, and we hope to do more in the future. This was number 1, so let's look forward to the next ones coming, and we'll look forward to seeing you all soon. Thank you so much.